Well, hello, everybody. If you're just joining us for this webinar, we're going to wait a couple of minutes until we're absolutely certain that all of you have joined us. So I apologize for this slight delay. Okay, hello, everybody. I see we've got now over 70 people have joined this webinar already. I'm just going to say you're all very welcome to this International Commission of Stratigraphy's celebration of 60 years of the International Union of Geosciences. We have a very varied panel. We've got a very varied group of talks covering almost the entire stratigraphical record and reaching into what's happening today and possibly into the future. We're facing a world with global heating, with sea level rises, with ocean acidification. We're looking at dramatic changes in weather patterns. We're looking at habitat loss. We're looking at biotic extinctions, all those sorts of things. So what we're going to try and do today is have a look at the past and look at some of the past extinction events and see if we can learn anything from those. And then we're going to move forward to the more recent past and what's happening today. And perhaps in the panel discussion led by my colleague Mike Benton, we might peer a little bit into the future. There's a couple of points I want to make. The talks are going to segue into each other, and so there won't be time for questions at the end of each talk. But what we're going to do is invite you to use the chat or the Q&A function to put your points there. And what we will do is we're going to pull together all these various questions and comments and save those for the actual panel discussions and move forward from there. To make it a little bit more interactive, we're also going to try and have a poll. And I think we have 5 questions that we're going to ask you all, and we'll put these up at the end of the talks, and we'll ask you to vote on your preferences for these 5 key questions. That should be quite exciting, and we'll read out the results at the end of the webinar. So what I'm going to do now is move forward a bit and share my screen. And here we go. Here you can see the list of talks and speakers today. The first series of talks we're going to run, as I said, back to back. And then we're all going to join together with questions from the chat and the Q&A for the panel discussion. I'm going to introduce myself now because I want to talk a little bit about the framework that we use to look at extinction events through deep time. And this is our international chronostratigraphic chart and how we got there and where we are today. Now, many of you will recognize this photo here on the left-hand side, possibly because you're all geologists and you know there are 2 global stratotype sections here in northern Spain at Zumaya in the Paleogene. Many others might recognize it from the Game of Thrones. And so it is a rather iconic way to start a lecture, looking at Cenozoic strata with GSSPs. Now then, I want to start right at the beginning and how our whole view of stratigraphy and chronostratigraphy actually works. We have to go right back to Leonardo da Vinci, who probably reinvented perspective in the hills of Lombardy and recognized that there were groups of strata that he could identify. There were groups of shell beds that followed on from each other, and there was already, through his perspective, some sort of order in strata and events occurring in the rocks that he was drawing. It was sometime later that Nicolaus Steno, the polymath Dane, who involved himself in many aspects of science and medicine and eventually religion, recognized in the Apennines that there was a definite order to strata. He called this the superposition of strata. And so the oldest rocks were on the bottom, the youngest were on the top. Sometime later, 100 years later, Giovanni Arduino and his colleagues, building on the work of Johann Lehmann and others, saw there was some sort of structure within mountain belts. The old metamorphic rocks were at the base, then there were cover sequences, and then there were consolidated rocks and sediments on top. And so not only could you order the strata from the oldest to the youngest, you could see some sort of structure, some sort of succession in mountain belts that would help us work out the order of geological events through time. And finally, William Smith, the canal builder, the English canal builder, in his work during the start of the Industrial Revolution in England and Wales and parts of Scotland and northern France and Belgium, was very keen on identifying particularly coal measures, but also transporting material around Britain along the canal network. He was building these canals, but also logging in the types of rocks that he saw. And he recognized that individual types of rocks were distinctive. There were shales, there were sandstones, there were limestones, but they also had characteristic fossils. And so as you go from North Wales with trilobites, Cambrian fossils of those sorts, you would go right through the whole gamut to the London Basin, the London clays with mammals and mollusks there. And so there was a way to correlate strata by using the evolution of life and particular fossil groups in particular rocks. And if we add the work of the great Scottish geologist, the founder of modern geology, James Hutton, we can add the whole time perspective, because up till then, people thought, well, tens of thousands of years old, possibly a bit older. But Hutton, with recognizing the three unconformities in Scotland, one in Loch Ranza and Arran, one in Jedburgh, but the most famous one at Sicker Point, looking at these vertical Silurian greywackies here, overlain by the near horizontal old red sandstone rocks here, little hammer for scale, indicated that these were the ruins of an ancient world under the unconformity. And so a lot of time had elapsed between the deposition and the folding of the Silurian rocks to the subsequent deposition of the old red sandstone, more alluvial type rocks above. And so Hutton was looking at a sort of ancient world, no vestige of a beginning, no prospect of an end. So there was a deep time going on. And others followed him and recognizing it was tens of millions of years, maybe hundreds of millions of years that we're actually talking about. Well, at the same time, geologists were working in Britain and on the continent to recognize distinctive groups of strata that they were going to call systems or geological periods in terms of geological time. And I've just picked out three here within the Mesozoic, the Triassic, the Jurassic, and the Cretaceous, just to give you a little bit of background about why we actually use these names today. The Triassic was named by Friedrich August von Alberti on a trio of rather distinctive rocks with red sandstones at the base, with limestones, with mussels or clams in the middle, and then the Kuiper marls at the top. And these were recognized in the southern part of Germany as a very distinctive package of rocks that von Alberti called the Triassic. Alexander von Humboldt, extremely famous naturalist, recognized limestones in the Swiss Jura that were, again, a very distinctive package of rocks, super limestones packed with ammonites. And he recognized that these were quite different from the Triassic, so he gave them the name Jurassic after the Jura Mountains. The problem was that von Humboldt reckoned they were older than the Triassic initially, but in fact, this was all sorted out and recognized that this was a younger group of rocks overlying the Triassic. And Jean Dallois, a Belgian geologist working in the Paris Basin, recognized chalk with these tiny coccoliths within it. And this was another very distinctive package of rocks that deserved a name, a sort of, broadly speaking, a type area, and with very distinctive fossils. And so we were starting to build up a whole succession of different rock systems that were being threaded together, rather piecemeal-like, to get our complete stratigraphic succession. Now, that wasn't too bad, because the succession at the time, the chronostratigraphy that was being developed, although it had various flaws in the way it was being constructed that I'll come back to later, it served the community very well. Georges Cuvier was able to use stratigraphy at that time to develop his ideas on catastrophes or mass extinctions. Charles Darwin was able to use the chronostratigraphy that was available at the time to develop his ideas about evolution through deep time. And I particularly mention John Phillips here because these are some of the first numerical curves of paleobiodiversity through deep time. And in his book, he actually uses numerical statistical data based on the ranges of fossil groups that were available at the time to construct this paleobiodiversity curve. And this was rather interesting because he already was able to note with some precision two of the most major extinction events that we see throughout geological time, one at the end of the Paleozoic and one at the end of the Mesozoic. And these extinction events were able to partition his curve into Paleozoic life, Mesozoic life, and Cenozoic life. Well, what was wrong with that? Well, nothing really, except a lot of these units were based on an idea of recognizing essentially a stage. They were unit strata types. And this is the Bajocian here, named after Bayeux, which is fairly close by, the beautiful sections on the northern coast of Normandy, where d'Orbigny was able to recognize, again, a package of rocks, which he called the Bajocian stage. They are defined in that you can recognize the package of rocks with their actual characteristic fossils, and we have a type area where these rocks crop out, and so we can actually use these unit strata types to build our stratigraphy. Unfortunately, as we began to realize, it's not a very good way to put things together, in that there are going to be overlaps between the different systems, and there's going to be gaps within the succession, and so it becomes very difficult to correlate these units very precisely across the globe if we're wanting to develop a whole framework to look at biological and geological events very precisely through deep time. And so we developed fairly recently, through the International Commission of Stratigraphy, the idea of global boundary strata types. And so the basis of these units, whether it's a system, whether it's a series, or whether it's a stage, are actually defined at the base. And so the next unit, or the top of a unit, is defined by the base of the succeeding unit. And this is how it works. It's fairly simple. Working groups, often involving many hundreds of geologists globally, will decide on the best possible section and the best possible means of correlating the base, and then come up with a proposal to show that we have this succession here. We should place the boundary strata type here within continuous sedimentation, and all sorts of other features are continuous through this that we can use to correlate. And then we stick in a golden spike. We're not a very rich commission, so the golden spikes are not usually gold, but we do mark the base of these units in some sort of way. That's a fact. Once defined in rock, we generally don't move these. What is interpretation is the way that we actually correlate the golden spike, or the bases of these sections. And this is in a little bit more detail. It's a more complex diagram, just to show how we can have a GSSP, a boundary strata type here, that we correlate across different sections. Unfortunately, there's a gap here, so it's very difficult to actually do that, if not impossible. When we're talking about rock, we talk about stages. And when we talk about time, this chunk of rock here represents an age, okay? So there is a differentiation between rock and time that's very clear. And some of the boundary strata types, they're all very different. Here's one in the Cambrian in China, the Wuliyuan stage, and this is marked here in this very well exposed succession, and it's marked by a agnostic trilobite, which we use to correlate it. It's defined here in rock and correlated with a trilobite. If we go now up succession into the Cretaceous and the Hauterivian stage within the La Cherche section and Drôme in southeastern France, a little bit different. Same idea, rock succession, put the spike in, except we're using an ammonite in this particular case to correlate it. And then finally, as we move up into the Holocene, it's not so fashionable to actually use rock because there isn't so much, but we use ice cores. And here's one of the ice cores from the Greenland store in central Greenland, where we recognize within the core on geochemical criteria, the base of the Greenlandian stage, for example. And perhaps even more wacky is this guy here, the Speleothem, which is our youngest of all the Phanerozoic stages. And it's marked right in here in a Speleothem, in a cave in India. And this marks the base of the Megalaean stage, which is also coincident with some major climate changes and with major effects on the civilization at that particular time. And so as it's a little bit strange, it's not typical of our other Phanerozoic boundaries, but nevertheless, that's where we are. And so we end up with our stratigraphical record here, the International Stratigraphic Chart with all the various golden spikes here. We can now calibrate this, and I'll not go into this in any great detail, but the radiogenic isotopes and our Milankovitch cyclicity are adding orbital tuning to this very precise chronostratigraphic record. And so we end up with a stage definition rather like that, very different from the original Bajosian stage that we saw, but with all sorts of different data here, enabling us to actually correlate the base of the Chattian and within the Chattian stage. And so I'm going to end it there. It is a global enterprise that we're involved with, involving many thousands of scientists throughout the world that are each working on their particular boundaries. About 80% of our GSSPs are now defined. We have now a fairly continuous record of geological time from the origin of the Earth to the present day. It's a great framework that can be calibrated and a primary focus for research, teaching, and outreach. So what I'm going to do now is end my talk there, and you're very welcome to put questions in the chat. And I'm going to hand over to my colleague, Mike Benton, who's going to move on and talk about the significance of these extinction events through time. So I shall try and stop sharing my screen and hand over to Mike. Thank you.
Great, thank you very much, David. That was fantastic. And I couldn't find the reactions on my Zoom screen, so I couldn't give you a round of applause, but thank you very much. So I continue with the question of why do we care about mass extinctions today? And this is very much part of the history of stratigraphy, the impact of IUGS, and I'll develop some of that a little bit in the talk. When we think about mass extinctions, what do we visualize? Is it this? Dinosaurs and objects flying through space, these dramatic illustrations, even more dramatic. I don't think you could generate anything more horrific than this kind of image. So what do we know, and how does stratigraphy help us? And here are some key questions that do we simply, as earth scientists, look at mass extinctions as individual weird events that are worth looking at just because of their strangeness? Are there any lessons for today? I'll touch on this, but we'll hear more about this from other speakers. Should we think about mass extinctions as purely negative, or can we identify positive aspects? And finally, of course, wrapping back into the overarching theme of how does our knowledge of precise rock dating help us in understanding these events? I'll start with the theme of biodiversity. This is a big topic that, of course, is common in discussion by all thoughtful citizens of the world. And we think of biodiversity, first of all, as the wonder of life. And of course, people study modern biodiversity to try to understand how many species there are, what they all do, how they relate to each other. But of course, we have a very immediate concern in almost all cases of what are the threats to biodiversity. So this is one approach, is to look at the relative proportions of species. And on Earth today, insects, shown on the right, by any count, make up the largest portion of biodiversity. I have to say, though, that this diagram does not include microorganisms. It's pretty much just the larger scale organisms. Actually, how many species are there on Earth today? Quite difficult to estimate. You would think that we would know this, and it's important for paleontologists and earth scientists to be aware of what we know about the present world when we try to interpret the past. And so something like 2 million species of plants and animals and fungi have been named. Maybe 300,000 of these are actually synonyms. So perhaps, but we don't have a complete list, there are something like 1.7 million named species that have a formal name. How many are there out there waiting to be named? Of course, there are estimates of millions or tens of millions, but I think most people today would say there are something like 10 or 15 million species, of which about 80% live on land, 20% live in the oceans. That's a very crude estimate. But we are concerned about rates of extinction at the present time. We are familiar with a particular species that went extinct historically, like the dodo, which is shown here, a kind of icon of regrettable extinction in historical time. And there have been many estimates of rates of extinction from these known historical extinction events. And however you plot it, for example, in the diagram here, the rate of extinction is rising. And the latest review, I haven't found a more recent one where a global effort has been made, is that the rate of extinction is demonstrably today much, much higher than what you would expect in the background. So I give a rough calculation here. If there are, say, 10 million species on Earth, if the average duration of a species is 1 million years, something that's about right, you would expect to see 10 natural extinctions each year. And of course, the number of extinctions by any estimate is considerably higher than that. Estimates of anything from 10 species going extinct per week up to 690. So a tremendous breadth of knowledge and estimates. And it's those kinds of estimates that have led people, biologists, philosophers, historians, to say we are in the midst of the 6th mass extinction today. And you can find many iconic diagrams. There are books such as Elizabeth Colbert's here and others, which use this expression 6th extinction. And I think most people know that that term 6th extinction comes from our understanding of the past mass extinctions, of which people have tended to identify 5. Although I think it's debatable that there are almost certainly more than 5 of these big extinction events. But anyway, these are the big 5 labeled here in this classic diagram, which represents, although it's got labeled age of reptiles, age of mammals, this is simply the diversity of life in the oceans. But it's the most well-known classic diagram from the work of Jack Sepkoski. There are the big 5. In today's session, we're going to be hearing about many of these, the end Ordovician, end Permian, end Cretaceous being 3 of the big ones we're going to hear about. And they've been characterized in many ways. This is a diagram by Budjan Lambeth that I just found by chance, which gives a very handy representation of the big 5, a suggestion of the scale of extinction, which at the species level, in these paleontological examples is very high. And I won't go through all of the discussion about what the causes are. But reasons to be cheerful. Well, we are survivors. We are here. If you are survivors, of course, then you could regard that mass extinction as positive. And each of those past mass extinctions, including additional extinction events somewhere near the end of the Precambrian, in the late Cambrian, as well as the others, have all been, I'm going ahead, characterized then by a following diversification of life. And shown in these different diagrams by myself, marine on the left and terrestrial on the right, we can see that, for example, the end Permian mass extinction event does punctuate the history of life in a very major way. And this was very nicely identified by Dick Bambach a number of years ago. He talked about the evolution of seafood. He was thinking, I think he was a man who enjoyed his marine fossils, but he also enjoyed a good meal. And so he postulated if chefs today only had Paleozoic organisms to offer, you would have brachiopods, trilobites, crinoids, and graptolites. Your platter, your seafood platter would be pretty poor. Whereas if you add the Triassic and Jurassic organisms, you go away having had a good meal. Because from the Triassic onwards, we identify a great diversification of gastropods and bivalves, of crustaceans, of various groups of less armored fishes, all of which represent an increase in energy, capture of energy by the system, increase in biomass, the amount of carbon that is locked up into the living system, and of course an increase in productivity, which links all that together, which is why these Mesozoic and subsequent organisms are just much meatier and much more delicious. That big change happened. And we've identified essentially a Triassic revolution, which is one of these positive outcomes, if you like. The end Permian mass extinction was the biggest of all time, but then it was followed by the biggest change in the whole of life. I'm not going to read through all of these, but it wasn't just new groups of organisms. They were doing totally new things. And this was called the Mesozoic Marine Revolution by Guérin-Vermeil. And similarly on land, there was a revolution where a whole bunch of organisms, including mammals and dinosaurs, became warm-blooded. They were capturing more energy, they were moving faster. It was a kind of predator-prey arms race. And a whole bunch of new organisms appeared throughout the Triassic in the same way as in the oceans, including new plant groups, new insect groups, and new vertebrate groups. We've seen this diagram already. Of course, this links to stratigraphy. It was John Phillips who noted this. This is one of his diagrams, but he did not read into it the idea of mass extinctions. And really, people were quite quiet about that for a long time. There is a very direct link, of course, in that the naming of these stratotypes, we've heard from David about the golden spike. This is a very famous one, of course, the Permian-Triassic boundary at Meishan in China. And it's a wonderful example to show how pinning the boundary to within a millimeter in a recognized section like this enables us then to begin to talk about the nature of a mass extinction in some detail. And the formal aspect, the international aspect that has been overseen by IUGS, the business of naming and labeling and being very precise and agreeing on an international scale, has gone hand in hand. It has been happening at the same time as massive improvements in the quality of dating, partly because radioisotopic dating is tuning to more and more precise dates. The error bars are much smaller than they used to be. When I was taught geology, the error on many radioisotopic dates was plus and minus 5%. Now the errors are fractions of 1%, so orders of magnitude improvement. But we mustn't forget, it's not just the geochemistry of dating that has improved, it's our linking that to cyclostratigraphy and being able to identify time units of 10,000 years, even 1,000 years, at any time in the geological past. And that now opens up some great possibilities. Growth of interest in mass extinctions has been quite recent. This rather ancient diagram I did showed that it was really only after about 1980 that people picked up interest. Before that, in a way, mass extinctions were not of interest. They didn't happen, or nobody cared about them particularly. This coincided with the Alvarez asteroid impact hypothesis, which everybody is aware of. And of course, there's been a to and fro of debate. Was it asteroid? Was it major volcanic eruption? And volcanic eruptions have dominated for the other extinction events. And I'm coming near the end. I'm going to draw together precision in stratigraphy and our ability to resolve these kinds of questions. Is it impact? Is it volcanism? It's impact. Beautiful study by Pincelli Hull and colleagues in 2020. and they showed in detail, because of advances in stratigraphy, they were able to generate this diagram at the top, which shows the KPG boundary highlighted within magnetostrat zone C29R. And they were able to show from oxygen isotopes accumulated from all over the world that temperature was, of course, fluctuating. And you can see a rise in temperature, then a fall at the KPG boundary, and then a kind of steadying of temperature afterwards. And the point of what they were able to do was they could discriminate two events, a warming event and a cooling event, separated by 200,000 years. To a geologist, that's a miracle. To an ecologist, that's pathetic. 200,000 years, that's ridiculous. But that really resolved, I believe, a very major question. And we know for sure, both things were happening, both had impacts on life. It was the impact that killed. And another beautiful piece of work in the same kind of precision dating, which we will hear more about from Melanie later, but connecting with this idea of improving the precision all the time. And Melanie will explain in more detail, of course, but the ability to look at fossils that are positioned at an exact point, and in this case, to look at the growth rings within the fossils to determine the time of year at which an impact or at which an event occurred, using a mix of precise dating, new methods in geochemistry, linking it to the seasonality and the growth of the organism to say that the meteorite hit in late spring. Wow. Nearly there. Improvements in precision and accuracy. We know that accuracy is important, precision is important, but we must be confident we have accuracy also. We constantly remind our students they have to be clear about the meaning of these two words in English. Precision is improving all the time, of course, as we reduce the error bars. Are we sure we're getting accuracy too? The combination, though, of isotopic and cyclostratigraphic methods, coupled with radioisotopic dating and international agreement about time zones, allows the potential to go even closer, maybe down to 500-year increments of time. So generalizing mass extinctions, what do we learn from the past that tells us something about the present? Despite the great interest in the KPG mass extinction, probably that's not helpful for our understanding of present threats, whereas many of the other mass extinctions, including the end Permian, are hyperthermal events, and they share a model. And this is something like the model that warming caused by excess carbon dioxide and methane and water vapor in the atmosphere, whether those gases come from volcanoes or human sources, can then lead to a whole series of consequences, including anoxia in the oceans, acid rain, acidification, loss of plants on land, loss of calcium carbonate, shelled organisms in the oceans. And that hyperthermal model now seems to be applicable to 10 or more past events that, and also probably to the present day. So again, I think that this is a very important consequence of our study of mass extinctions. Yes, it does have relevance to the present day. And so I round up here with a summary that the common elements of many mass extinctions have come to the fore recently. They are individual events, but some of them, at least, except for the KPG event, may share common characteristics. There are lessons for today. Not only are we experiencing the sixth mass extinction, but it may be that the current event can be classified as a hyperthermal, meaning we can make certain predictions about what might happen. And modern methods in dating, including international agreement about time zones, have been enormously important in enabling us within a few decades to massively improve the precision of what we do. And so the dinosaur is happy because we have this new understanding, and I thank you for your attention.
Okay, thanks, Mike. I think we're all happy. That was a very comprehensive talk that really sets the scene for all subsequent speakers on this call. So thank you very much for that. I think I'm going to be next. Cameron Penn Clark was going to present on the Devonian, but he had to drop out at very short notice. So I'm afraid you're going to have to listen to me talking about something I do actually like very much, and that's the end of the Ordovician extinction event. So I'm going to pop on now and share my screen, and I will put it up to the little bit tricky here. Should be all right. There we go. So what I'm going to talk about is the end of the Ordovician. And so it's very interesting from a number of points of view. I think the first one is that it's the first of the big 5 extinctions, and it's the first extinction that actually involves sort of big organisms, animals and those sorts of things. And so we're looking at a metazoan extinction for the first time. The second point I'd make is it was very short, and it's associated with a very major ice age. And for those of you working throughout the stratigraphic column, you'll say, well, ice ages don't usually drive extinctions, and that's absolutely right. And so this is a little bit special in that the extinctions seem to be associated with the waxing and the waning of a very major ice sheet at that particular time. And so there are still a lot of questions to be answered about what actually happened at the end of the Ordovician. Okay, well, we know a lot about the major events in the history of Earth and life, and Mike has talked a little bit about them, right from the time at the very beginning of our planet, when potentially there must have been a single prokaryotic cell, right up to the present day, where we have over 20 million eukaryotes, complex organisms on the planet. And depending on how these estimates are calculated, possibly many, many more, and many more if we think about microbes as well. And so we have a very long record from the first evidence from carbon isotopes right back in the oldest sedimentary rocks in the Isua complex in West Greenland, right up to the present day. But the trajectory hasn't been smooth because, as Mike has pointed out in some detail, that trajectory has been punctuated by quite a number of extinction events. And so if we look at this diagram again in a slightly different mode, but it is an extremely important and useful diagram, you can see the 5 big extinction events here, going from the end Ordovician, the Devonian, the big one at the end of the Permian, Triassic, and the end Cretaceous, and we're going to hear a bit more about 2 of those later on. And they partition up very distinctive types of marine faunas, dominated here by trilobites and early mollusks and echinoderms. The Paleozoic evolutionary fauna dominated by filter-feeding organisms such as the brachiopods. Unfortunately, yes, there's not much meat in a brachiopod. And then on to the modern fauna dominated by mollusks and on land various initially dinosaurs and then mammals and those sorts of things. But this is the guy we're going to look at now, the end Ordovician extinction event, and see if we can learn a little bit about it and move towards possible causes and consequences. This is an old diagram. We know a lot about this. Databases change throughout time, but the positions of these extinction events more or less stay in the same place, although the amplitude does vary as we gather more and more data. There's a lot of excitement going on in the Cambrian, but this is probably due to the very small sample sizes that we have from Cambrian faunas, where a lot of these things are very much enhanced when we look at proportions. End of the Ordovician, end Permian, end Cretaceous, these are standout events when we look at the proportion of genus extinctions. If we home in now a bit more on the actual Ordovician itself, and here we've got the lower Paleozoic systems here, early Paleozoic periods. We got the Ordovician here, and we've got sort of various measures of biodiversity here with data taken from the paleobiology database. Not a very sophisticated treatment of the data, but I think it makes a major point here that at the end of the Ordovician, we see quite a major drop in biodiversity. We also see changes in the strontium ratios. We also see a major spike in the carbon isotopes, but I would link this quite importantly here to drops in sea level at this particular time. So these are different ways of looking at data through the system. The first thing I'd mention is what's happening with numbers of species and number of genera through this particular event. Well, yes, it is a big taxonomic extinction. Some people have estimated that about 75% of species actually disappeared here right at the end of the Ordovician. And this is following a very major diversification. In fact, one of the possibly the biggest diversification of marine life on the planet, the Great Ordovician Biodiversification Event, where we can see the plankton, the benthos, and then reef-building organisms all contributed to this hike in biodiversity in deep time. But that ended at the end of the Ordovician with this very major extinction event. And so a lot of species and genera do actually disappear. So it is a taxonomic extinction. Where we're less certain is it a phylogenetic extinction? And this is some work I did a while ago, looking at the phylogeny of some of the major groups of brachiopods. And as you probably can see, here is the late Ordovician mass extinction, the Lomi, through here. And yes, some major groups, and we have a sort of a genus here to stand for the whole family, but you can see some are actually disappearing in the rhynchonelids and in the strophomonates here. But some of the others are in the inarticulates. Some of the spire bearers are continuing through. So it's not such a big phylogenetic extinction as a taxonomic extinction. And then thirdly, if we look at the ecological extinction, well, George McGee, Mary Droser, Peter Sheehan, and others many years ago pointed out that unlike some very big extinctions, the ecosystem is not really shaken up, destroyed, and has to be rebuilt after the extinction event. We're seeing things at what they call the category 2B level, that we're seeing sort of community reorganization. So it's as if you know, you have a house and you've got a whole lot of 1960s, 1970s furniture. It's all very functional. It doesn't look very nice to us nowadays. You decide to throw out that furniture and go to IKEA and buy a whole set of new furniture. Okay, well, the same functions are being carried out, but your house looks very different. And so that's a little bit like the community reorganization that's actually going on in this event. And here we can see ecological severity is right down at the bottom of the list here. So probably not such an important ecological event. We've worked with a number of people over the years, and this is with Seth Finnegan and my former PhD student, Christian Rasmussen, and we tried to look at a sort of Ordovician brachipod risk register, looking at the ranges of brachiopods through the event. And the standout for us really was that some of these guys here, the deep water faunas that had been around for a very long time, disappeared at the end of the Ordovician. This was quite surprising because you expect stability in the deep seas, and a lot of these little guys seem to disappear at the end of the Ordovician, together with more familiar guys in the shallow water, in the warm water environments that you would expect to go during an ice age. So what actually happened Well, during the end Ordovician, we go from the late Catian stage or age into the early Hernantian and the late Hernantian, and the faunas change quite dramatically within the brachiopods that were the most common organisms around at the time. The deep water faunas, the Foliamina faunas, all these wonderful little guys that dominated the deep shelf and upper slope during much of the Ordovician, they were wiped out. So too, during this phase of spread of this major ice sheet, where a lot of those brachiopods that were living in the tropics and the subtropics and the shallow water environments, they went as well. And sweeping around the world, we see what's known as the Hernantia brachiopod fauna, dominated by half a dozen to 10 genera that are very widespread. They're almost the only show in town in terms of the brachiopod faunas during the early part of the Hernantian. And then as the ice sheet disappeared again, and we get flooding, we get a lot of anoxia, those sorts of things, the brachiopods appear back again on the scene in a slightly different guise in terms of the Cathay Edgewood faunas that creep back in various parts of the world in the tropics and spread out across the globe. So if we go right down to the very basic level of looking at individual taxa, we can track this extinction event. So what was happening? Is there a model? Is there some way of trying to understand how this rather sudden appearance and disappearance of a major ice sheet, and now we think it's in terms of hundreds of thousands of years rather than millions of years that we thought before? Well, first, the ice sheet does actually expand, and this involves a shallowing of the world's oceans. And here you can see Titanic is going to struggle a little bit as you actually draw down the sea level. And the species area effect would suggest that there's less space around, and our communities are going to be in the upper part of the slope and shallow water if we remove 100 to 150 meters of water during this event. The surface waters would cool down substantially, and so we would get this sort of situation here. In the first strike of the end Ordovician extinction, ice sheet retreating, the glacially enhanced productivity taking place as more components are eroded and come into the basin. But then things warm up again, and we see a rise in sea level, we see rising anoxia and those sorts of things. So in the early stages of the event, yes, the shelf is hit rather hard, but we also think with increased circulation in the deeper waters with the ice age, that a lot of anoxic, poorly ill-conditioned waters are coming up into deeper water environments, wiping out our Foliamina fauna. So it's still quite a complex event. Okay, why didn't it have such a big effect, and why don't major glaciations cause big extinctions throughout geological time? Well, this is rather an old paper by Brian Turner, Howard Armstrong, and others, pointing out that, well, actually, if you look at the Gondwanan margins, there's been a whole series of major glaciations throughout geological time, and we're picking up evidence for some of them in Baltuscandia here that had effects on the diversifications or extinctions of the fauna, and here's one at the top. And could it be that biotic resilience was built up during these phases of glaciation? And this one is quite a short one right at the end, but was very intense and probably caused our taxonomic extinction. Well, if I can summarize very briefly, we haven't talked about this today, but the main interval of bodybuilding took place during the extended Cambrian explosion in early to mid-Cambrian, but we see massive increases in diversity at species, generic, familial levels during the Great Ordovician biodiversification. But this short, intense glaciation was coincident with the extinctions and probably was a major cause of those in shallower warm waters and in deep waters too. Did the sequential increases in severity of global cooling enhance the resilience of the marine fauna? Well, that's an open question. And what were the causes of the causes? What kicked in to cause this major ice age and the associated extinctions? More and more evidence has accumulated for widespread volcanicity during the later part of the Ordovician. So can this be a trigger? I think standing back for a minute and looking at the early Paleozoic as a whole, we do see this trajectory capturing different patterns and processes as biodiversity is enhanced during this period of time. And certainly they may not have been good to eat, but the marine benthos of brachiopods and others survived for the next 200 million years until they really suffered pretty badly at the end of the Permian. Okay, so thank you very much for that. I'm going to stop sharing my screen here if I can. Yep, I've got it there. And I'm going to turn on now to our next speaker, who's Sen Xu. And you're going to talk a little bit about some of the very exciting changes in vegetation across the big extinction, the Permian Triassic event. So over to you now.
Okay. No, not this one. Sorry. Yeah, that should be all right. Okay, thank you for having me here. I'm Jen. Today, I'd like to introduce the story of the land plant during Permian Triassic. So the end Permian mass extinction about 250 million years ago saw the most significant drop in the plant species diversity. So since plant landing, the early Triassic has had the highest deforestation level. The land plant crisis left the world with over 5 million years of coal gap. No coal was preserved at all. And the super greenhouse condition in the early Triassic with the land temperature over 42 degrees might remind you of the hot summer we just suffered from. You may think that the most important primary producer on land, the plant, may have something to do with this abnormal condition, but we don't know for sure. Even the most basic question, whether plant has extinction or not, nor the evolution details are still remains unknown. So today's talk, I will go with two parts. First is the plant extinction and the recovery pattern during the Permian Triassic. Then we hire an Earth system circulation model to test the power of the plant. Here is how the Earth looks like during the Permian Triassic. The South China area is in the equator and has the marine GSSP. The chronostratigraphic resolution in South China is also the highest. So we will start the journey from South China first and use it as a standard, then explore other basins. So study the deep time plant assemblage could be concluded into 3 steps. First, we need to do the strata correlation to get the age of the flora, so we can compare and conclude flora of the same age to eliminate the influence of preservation. So here is the terrestrial Chahe section in South China. Each of the black line tells the distribution of plant species in the strata. We have a working age of 252 million years in bed 68, older than the marine Permian Triassic boundary, and most plant fossils disappeared in bed 69, and so is the last coal line. So we think the Permian Triassic boundary should be somewhere above the bed 68. We named the possible strata as Permian Triassic transition. Normally, the land strata is hard to compare due to the lack of fossils with reliable age. But in South China, we have abundant parallel sections. which contains marine biota and the land plant fossil together. For example, with the bivalve and the Conchostraca fossils, we realized that the herbaceous lycopode Tomiostrobus in South China is within the Permian-Triassic transition. So those lycopods could be used as strata correlation in other terrestrial strata without that biota. And considering the resolution of the time, the geochemistry record seems to be a good choice. The signals can be explained by many things. So we need multiple proxies to locate the event. For example, the wildfire proxies, the amount of the fire, like charcoal, we think is controlled by the amount of plant. And in the parallel Chinahe section, we can see the running out of the land plant before the marine extinction event. So we think the land plant extinction is earlier than the marine extinction. So after we correlated the strata, we need to normalize the fossils. Plant fossils is normally preserved fragmented, and each organ has a morphological name. We only need one of the most diverse or representative species to represent the whole plant, like the trunk of the Lepidodendron, and delete all the other duplications, like sporophyll, the fertile leaf, cohen, or roots. Here is the chronostratigraphic table in South China, and the gray part shows the flora's location, and the right part is the extinction and the origination curves in South China. And we can see that there is a clear increase of the extinction and decrease of the origination, which happened since late Permian, but has the highest peak in the end Permian. So combine the plant macrofossils, spore and pollen data, and the biomarker data. The plant evolution during Permian-Triassic could be concluded into 4 stages. Firstly, the extinction of the Gigantophthorus flora at the end Permian. Secondly, the survival and the pioneer of those herbaceous lycopods, Tomiostrobus in Permian-Triassic transition, followed by the diversity recovery of conifer-dominant flora in the early Triassic, then the biomass recovery, and finally, the land ecosystem is totally recovered in the Middle Triassic. So if we look at the middle latitude area, including the North China, Europe, and North America, where the aridity influence is stronger, the strata correlation is like this. Based on the magnetostratigraphy, zircon age, biota, and the carbon isotope. So during the Permian, the middle latitude area was occupied by those conifers, indicating the transfer from Paleophyta to Mesophyta already happened in late Permian in middle latitude area. And after the herbaceous lycopode zone in the Permian-Triassic transition, then in the Middle Triassic, those areas were all occupied by conifer again, like low latitude of South China. And we think those Triassic Wattia conifer is thought to be inherited from those end Permian. So in the high latitude area, like the North Polar Russia Siberian area, Middle Asia, and China Xinjiang, in Siberian, the extinction of lowland Cotaites happened in the late Permian, and there was no further extinction during the Permian-Triassic transition, but the herbaceous lycopode was common, showing Siberian has also been influenced. While in Middle Asia and the China Xinjiang area, we saw both the decline of Cotaites in late Permian and the extinction near the Permian-Triassic boundary. And in South Hemisphere, like South Africa, Argentina, Antarctic, and Australia, we saw the extinction of the Glossopteris flora. But in Argentina, the extinction of Glossopteris happened in the late Permian. And although there's no fossil preserved in the early Triassic, the Middle Triassic taxa was quite similar to those in the Permian refuge. So, unlike other places, there seems to be no extinction in Argentina during the Permian-Triassic transition. So we could see that the plant story in different latitudes is quite varied. And thanks to international cooperation in chronostratigraphic, we could see more details of global scale vegetation evolution. So those areas were arranged from north to south, from high latitude to low latitude. We identified there are two different events happening in the End-Permian. One is the transfer from paleophyta, which is dominant by spore plant and shown in the green color, and the transfer to mesophyta, which were dominant by seed plant and shown in the yellow color. So we could see these events were happening earlier in the late Permian and show as a extinction level, while in those low latitude area, there were more like gradual change to mesophyta. And another event was the End-Permian plant crisis, happened in all the low to middle latitude area at about the same time in the End-Permian, which is signed by the end of coal and spreading of Tomiostrobius. While in high latitude area like North Polar Russia and South Argentina, seems normal alternation of plant rather than a extinction event. So we calculated the extinction rate in different latitude. With the normalized data, we think the plant extinction only happened in low latitude, with the extinction rate up to 86%. So we can see that they do have some bad omens before the big events happening. So here is the result of the global plant fossil occurrence with the land tetrapod shown in the black color and the vegetation reconstructed based on the sedimentology and the fossil record. And here is reconstructed plant primary productivity. So we can see there is a massive loss of land biomass during the early Triassic in the low to middle latitude area. To test the power of plant, we hired a three-dimensional biogeochemistry climate model to do that. So here is our result. We set two scenarios. One is the green shadow line, shows the result of vegetation and the volcanism working together, and the black dashed line shows the scenario without the vegetation. And it's always the scenario with vegetation and volcanism working together that fits the best with the geological record. So we proved that the 5 million years of early Triassic late solid hot was caused by land plant collapse. And after the plant recovery in middle Triassic, the Earth system returns to be a good place to live. So we suggested that thresholds do exist in the climate carbon system, and beyond which the warming may be amplified substantially. To give people a vivid impression of what's happening during that time, we cooperate with our art school, Huisu Studio, to make the video. So let's time travel back to 250 million years ago. Sorry, that takes a while. Oh, sorry. So that's all. Thank you all for coming, and also thanks for IUGS and our team members.
Okay, well, thanks very much for that. Some really striking images to end your presentation with, and a most striking event. So what we're going to do now is I'm just going to put up the program again to let people know where we are. And well, going to see a little bit of Antarctica by mistake there. Let me just pull that in here. And what we're going to do now is turn to another of the very high profile and major extinction events, but one that we know a huge amount about and much more about due to Melanie's work on the actual seasons in which this major event actually occurred. So I'll turn the screen over to Melanie now to talk about this apocalypse at the KPG boundary.
Sorry, I first had to unmute. It wasn't the right order. Okay, let me see. Am I full screen now? Yes, okay. So I'll be very honest. This was actually my original title for my paper, but my co-author Jan Smit didn't like the term apocalypse. He thought it was too biblical. So I use it for my talks now. So I'm going to take you through the most recent, largest catastrophe that decided the trajectory of life on Earth. And I look forward to having discussions afterwards. So as already mentioned by both David and by Michael, the KPG extinction is quite different from the others because it is such a sudden, catastrophic extinction. It's actually so sudden that we can bring it back to a single day. And because we could bring it back to a single day, there were some methods that I could apply that are otherwise not possible to apply. And so I tried to reconstruct the season of the impact. And I will take you through these methods and the results and show you how I got there and also why this matters. So I cannot talk about the KPG extinction without raising the Deccan traps. It's an ongoing debate still. And of course, there was a large period of volcanism at the end of the Cretaceous. But there also definitely was a meteorite impact. And as Mike already showed, this coincides with the actual extinction. But of course, it could be both. As Chiarenza in 2020 also showed, there's been a lot of arguments always about this 3-meter gap. Were there even dinosaurs at the end of the Cretaceous? Because for a while, people struggled to find them. But in 2020, Chiarenza et al. set them straight and showed that the biodiversity of dinosaurs, they were not struggling. They were thriving. They were thriving until they thrived no more, which was when a giant asteroid plummeted into Earth. And so it was sudden and it was without warning. And this meteorite, it struck Earth, and it's like you throw a bowling ball into a sandbox. Liquid pieces of Earth flew back up into the atmosphere and well above it. Some may have even circled the moon. And these impact spherules or tektites later on rained back down on Earth, and some very clever geophysicists have calculated that this fallback time was it started between 15 to 30 minutes after the impact. And you can find these little impact spherules everywhere if they are preserved, just below the iridium anomaly marking the end of the Cretaceous. This asteroid was roughly 12 kilometers in diameter and caused an extinction with an extinction rate of roughly 76%. And it's a quite selective one, although of course this is dependent on how you look at it. When it comes to biodiversity, the biodiversity recovered quite quickly after this extinction, but it was filled with a lot of different a lot of different organisms, mostly from the mammalian groups and not necessarily from the dinosaur groups anymore. So we can bring this extinction to the last day. And because we can bring it back to the last day, it means we can study when this day took place. And we can do that in Tanis, which is a locality in North Dakota, actually the most southwestern corner of North Dakota. And we can do that by studying a seiche deposit. And a seiche, I will explain in a minute, but it's a deposit that covers a point bar almost like a blanket. It's really like a giant wave went over it and laid itself down there. It is quite far from the crater, although you could of course be much further, but at 3,000 kilometers distance, I don't think we can ignore the fact that that's quite far. So a seiche deposit is a deposit, I'm just going to turn off the sound because it's not necessary. It's similar to a pool during an earthquake. What happens is that the seismic shock wave goes through the Earth's crust, and it doesn't shake the crust necessarily as much, but it does definitely shake the overlying bodies of water. And what you then can get are waves at like 30 meters height, coming over North Dakota and burying everything in its path. It took with it material from the Western Interior Seaway, and it mixed it with freshwater materials and terrestrial materials, while the tektites or impact spherules rained down. And here at the top, I can actually use the laser pointer, I think. You can actually see that the sediment structures are deformed, and at the bottom of this is an impact spherule. And we can find them like this. We can find them in little lenses in the deposit, but we can also find them in the fishes. And so I went to Tanis and I excavated these fishes, and you can see these fishes are deposited in the direction of flow, which is really the most phenomenal thing I've ever excavated, because in the lower part you can see all the fishes in this direction, and a little bit above you can see them in the opposite, and in the opposite, and in the opposite. It's mind-blowing. And these fishes have impact spherules in their gills, which confirms that they died on the day, which is great news because these fishes are ectotherms and they grow in annual growth increments. So another picture of these gorgeous fishes. So my research question was, can the fossils from Tanis tell us in which season the meteorite impacted? And for this, I used one paddlefish skull to confirm that they had indeed died on the day. And I collected 3 paddlefish jaws or dentaries and 3 pectoral fin spines from sturgeons, or breast fin spines, whichever way you want to call them. And I cut them into thin sections. I applied X-ray fluorescence. I sampled the growth increments, and I used stable isotopic analyses, not to date them, but to assess their environmental conditions and whether they were growing or not, or eating actually or not. And then I applied synchrotron tomography to study the bone cells and to locate the impact spherules in the gills of the paddlefish. So growth marks in bones, in certain fishes, not all bones grow like this, but these fishes, they do not stop growing. They keep growing throughout their lives, which means they record every year that they live, which is a real source of information. And these fishes have certain bones, the dermal bones, for instance, and they grow every year without remodeling the previous years. In some cases, the inner bone gets resorbed and then deposited on the outside, but that's not the case with these fishes. They grow every year like a tree, pretty much. So similar to these rings, we can study them, and it's not as simple as light and dark, but what we have is that we have a phase where osteocytes or bone cells are being produced, which is the growing phase. And then when growth slows down, that's when you get an annulus, you get this laminar growth, and then growth stops. And then as the new year begins, you get new growth, you get new osteocytes, and we'll call them bone cells for ease now, but they get bigger when growth is fastest, when consumption is highest. So for instance, in this fish, you have an example of 6 years. There's a vascular canal that can distract a little bit, but if you can follow it, then you know it's a canal. But this is an example. And here, when the fish dies, documents, in this case, the last day. So we applied X-ray fluorescence, and X-ray fluorescence can record the elements that are present in the bones or in the rock. If it's like in this case, we have 2 dentaries next to each other with a lot of rock in between. And this allows us to assess the degree of elemental exchange between bone and rock, or diagenesis. And if we want to do isotopic analysis like carbon and oxygen, then we want to know, are we measuring the original material, or are we measuring the result of rock formation, of diagenesis? And this was quite relevant for that. And then we did the stable isotope analysis. And for this, I took a micromill, and here you can see a scale bar. This is 0.5 millimeter. And I drilled out the growth increments in this paddlefish dentary or jaw. And I made individual samples of these for isotopic analysis. And we measure the ratio of carbon-13 over carbon-12. Carbon-13 is the more rare isotope, and carbon-13 increases with the food chain. So autotrophs, or that are the primary producers, or like the plants you just saw, they prefer to use carbon-12 to make sugar, and carbon-13 is apparently not as suitable for that. So they use that to put it into their exoskeleton or in their plant structures. And so anything eating that therefore gets enhanced in carbon-13. And the higher up in the food chain you go, the more carbon-13 there is. And when it comes to the oxygen isotopes, oxygen-18 is the heavier oxygen, and oxygen-16 is the lighter oxygen. And therefore, you have a preference when it comes to precipitation and evaporation. The lighter one will more easily evaporate, and the heavier one will more easily precipitate. So we went to the ESRF, the synchrotron in the European synchrotron in France, and we scanned this partial paddlefish, which is because it was partially prepared, as you can see, a lot of the rock was already removed. It was a very fragile specimen, so we had to wrap it up in a lot of styrofoam and then load it into a tube. We put the tube on the sample stage, and then we fired X-rays at it while we took pictures, which is similar to getting your CT in the hospital, but then this one you won't survive as a person, so it's a good thing we only use it on fossils. So the results. So looking at the thin sections, this is 5 of the 6th, we could immediately see that we had the presence of the lines of arrested growth, which is exactly on the intersection of when growth stops and when growth begins again. So you could roughly call that winter for ease. And we see this trend in all these individual, so here we have a sturgeon, a sturgeon, a sturgeon, and then we have 2 paddlefishes, and they're all roughly the same age, between 6 and 8 years old. And am I moving on? Yes. So, and here we have the results of X-ray fluorescence. And this can look like a very confusing image, but I will walk you through it. For instance, we would like to know if elements like potassium, silica, manganese, and iron have moved in the fossil, or have come from the rock and entered the fossil matrix, the fossil bone matrix, but they haven't. They've actually dominated in the sediment, which you can very clearly see in the small bits of rock that have remained on the sides of these bones and in between these two in particular. But when we look at the calcium and the phosphate, then we see a pattern of very clear, consistent preservation in the bone. The preservation is really quite good. Last little detail, what we see is that iron here in green and manganese here in blue, which are both elements that are very occur in blood, that they've been preserved inside the blood vessels, which is unsurprising, but it's nice to see that they sort of oxidized there. And then we did the synchrotron tomography. So here we have a fish, and I'm going to play a video of the fish in a minute. But the first thing we noticed is that inside its skull, we have the preservation of a brain case, which also confirms that the preservation of the material from Tanis is really good. It's like they died in place, got buried there alive, and nothing happened to it. This is ongoing work, but I'm trying to get this brain case out and study it. It's quite a task. And then we look at the tektites or impact spherules in the gills, and we find these perfectly spherical structures. Some of them are a bit broken. I'll circle them for ease. But you can see them next to these beautiful little gill rakers. You can see these. They don't really have a contrast because they're made of roughly the same material, or at least the material absorbs X-rays similar to the deposit. But they're so spherical. It's almost, once you see one, you'll see all of them. So here is the little paddlefish that we scanned. I put some googly eyes in there so you know where the front is, because otherwise it's a bit hard to see. And then we move to the gill region, and then we see in a higher resolution scan, we see that behind the gill cover are, of course, the gill arches with on there the little stubby gill rakers. And between those gill rakers, we find these spherical, I made them yellow here for ease, impact spherules. And they're all between the second and the third gill arch, which may tell us something about the feeding pattern of this fish, but I've not figured that out yet. And they're all perfectly hollow on the inside, because they formed in space. One of them actually preserved some unaltered material from the Yucatan Peninsula, but I can only reach this digitally. If I would want to study this material, I will have to cut open the fish, which I don't think they'll let me do. So then we read the growth lines, and we do this because I also used synchrotron scanning on these little sections of bones. So just below where I cut this section, I could actually also use synchrotron scanning at a 4 micron resolution on these same sections of bones. And when you think of this resolution, when I say it, it's not just the optical resolution of a single image, but I could actually also scroll through this, and I could stack multiple slices on top of each other to find these osteocytes or bone cells, which are the darker bits here. And what we find is if we combine these red arrows, which are the lines of arrested growth, which are winters, and we combine these green arrows, which are the highest numbers of bone cell volume and density, is that they, of course, perfectly alternate. Winters and summers are their opposites. And what we see here is that when we look at the time of death, which is always this edge, then we are just after a winter, but we are not yet near a summer. But that was not sufficient for me. I wanted to be able to quantify the bone cells. And one of the reviewers of our article suggested that we do so. The only thing is that the method that we apply for this is aimed at a resolution 10 times higher than what we had. So this it's qualitative quantity. So it's don't take the exact numbers for it, but take them as indicative. And what we see is that the amount of bone cells and their volumes, here again, we have the maxima or summers, winter, summer, winter, summer, winter, summer, winter, summer, winter, summer, winter, summer, winter, spring. I'm going to take a sip. And then we have the stable isotope analysis. And what we find in these stable isotope analyses are in oxygen likely patterns of migration, which this fish didn't really do. What we think happened is probably that there was influxes of other fresh waters in the river that it was swimming in, because it's a paddlefish, and the paddlefish does not migrate between freshwater and marine. And this also confirms that, because otherwise it would be spiking all over the place. And the carbon, it also doesn't fluctuate that much, but then again, this paddlefish is also not very high in the food chain. It's a filter feeder. It's not an apex predator. But what if we combine this data? And that's when we get the graph that Mike already showed earlier. And that's when you see that things actually add up. When carbon isotopes increase, when consumption increases, that's actually also when the osteocyte production is highest, which means that it sort of makes sense to me when I put this all together, but it actually means that when a fish eats, it also immediately grows. And this is something that it makes sense in your head, but it also helps if you actually have the data to prove this. And in this case, this proves that this fish died after a winter while it was on its way to while it was on its way to summer, but that summer never came. So conclusions: on a spring day, 66 million years ago, the Cretaceous came to an end, to a halting stop. And then it's at least 30 years of cold and darkness. So why does the season matter? Well, if it was spring in the Northern Hemisphere, then this means it was autumn in the Southern Hemisphere. The opposite. There's no other way. So what are then the implications? Well, spring is the season of growth. It's when plants grow new leaves. It's when animals go outside, they tend to their offspring, or they make offspring, they look for food. Whereas in autumn, organisms actually prepare for winter. so they prepare for their winter sleep. They prepare for their resting phase. And compare a plant in spring, when it suddenly freezes, they die. Whereas in autumn, they're generally speaking, they're fine. And furthermore, the recovery was over twice as fast in the southern hemisphere than in the northern hemisphere. So I'm going to leave you there and we can discuss it later because I'm going over time, I think. Thank you very much.
No, thank you. That was absolutely incredible. So that was absolutely wonderful. We're going to move on up the stratigraphic column now, and we're going to move on to Alina Lawrenson, who's going to talk about these big guys up there in the Pleistocene megafauna. So over to you, Alina.
Thank you very much. Ah, Melanie, man, that's so crazy. All of that information about a day 66 million years ago, that is quite striking. Thank you very much. First of all, Dave, thank you so much for putting together this this panel of really wonderful talks. And I'm thinking that if I'd only heard these talks 15 years ago, my trajectory would have been much easier because I'm a biologist by training. And I have to say, rather late did I understand. We didn't have any geology or paleosciences when I went to university. And I think I really only understood rather late how important it is to understand the concept of time and what has come before, both from the geosciences as well as from the biosciences, in order to understand where we are today. So thank you very much. This has been, I've already learned rather a lot. Now I'm going to try and share my screen here with the right screen. Here we go. All right, can you see my, whoopsie, oh shit, okay. I'm going to steal my thunder and just zip through all of my slides here. Sorry about that. Okay, here we go. All right, so I'm going to talk about the most recent. So now we're going to zoom forward 66 million years ago to the late Quaternary. So this is the around the last 50 to 100,000 years, during which there's been a massive turnover in fauna, so in the animals that we observe in the landscape, not only in the species so this is a period during which a lot of species disappeared. If we step back even 15,000 years, we're going to be knee-deep in biodiversity. And we would see species in the landscape. Even here, I'm sitting in Denmark, where there were mammoths, there were woolly rhino, there were wild horses, there were aurochs, species that have gone globally extinct today. And this was something that happened across the globe. It's associated, or rather this happened, so a lot of this has happened within the last 50,000 years. In some regions on the planet, it started a little bit earlier, but it's within around the last 50,000 years that really we've seen this massive turnover in species loss, but also in species composition, where the species that we see together today, when we go back 5,000, 10,000 or 20,000 years, the species that we would see together in the landscape are completely different from the compositions that we see today. So this is actually going back to an earlier talk. This is from one of the Greenland ice cores, looking at which we use as a proxy for temperature. So this shows the changes in temperature across the last 50,000 years, where, let me get my pointer out. Whoops, how do I do this? Whoops. Laser pointer, here we go. Where in a particular, when we go from the last glacial, then we have the last glacial maximum from about 23 to 18,000 years ago here, depending on where we are on the planet. And then we move into our current interglacial, so into the Holocene. And in particular, massive climatic upheaval at the interchange between the last glacial and our current interglacial. So at the interchange between the Pleistocene and the Holocene. And this is also the period of time where we see mass species extinctions. So just walking through the extinctions at a global scale. And across the continents, actually the number of genera, which we use as a proxy for the number of species, so the number of genera that went extinct were actually rather different. So if we look at some areas such as Africa, only really we see 18% extinction of, and these I should say, so I'm talking about, we've heard about plants and we've heard about fishes. Here I'm talking about large mammals. We call these megafauna, and they're arbitrarily set at big mammals over 45 kilos, so something like a big Alsatian, anything bigger than that. And this shows how large a component of those species were lost over the last 50,000 years, so during the late Quaternary. But it also shows a big difference. So in North America, we see, almost 3/4 of species that are lost from the fossil record. In South America, even more, over 8 in 10 species, or genera in this case, but as a proxy for species, go extinct. And also Australia, almost 90% of species lost within a time frame of only a couple of tens of thousands of years. So an incredible species turnover. Interestingly, we do have a couple of continents where it doesn't look quite as bad. Africa, only not even 20%, and Eurasia, about 1/3 of the species are lost. Now, when we look at the size of the species that were lost, these show each of these plots is from a geographic region, and where we see really that the species extinctions during the Pleistocene, so these are just going into the Holocene around 11,000 years ago, it's all the big species that are lost. Again, we have these where we saw Australia massive extinctions, also all the big species are lost. Similarly, in South America, all of them are lost. In Africa, this is the only place where we still have all categories of species sizes left. So there's some interesting biogeographic patterns in where we're seeing large-scale species extinctions on some geographic regions relative to others, not only in the amount of species, so in the magnitude of species lost, but also in what kinds of species are still left in the landscape, where Africa is really the only area where we still have representation of the biggest species alive. Now, interestingly, the pattern of decline and where we see the largest magnitude mirrors very closely the biogeographic history of our own species and of other of our human-like, not ancestors, but some of the human-like groups, the humanoids that were present in the landscape before modern humans came out of Africa. So this plot here shows you the biogeographic scenario of modern humans originating in Africa, moving into Eurasia, and then at the opening of the Bering Land Bridge at the onset of the Holocene around 15,000 years ago, we were able to push into North America and then subsequently move south into South America. Another trajectory was after we moved into the Middle East, that modern human populations moved into South Asia and then into Australasia. Now, if we, on these geographic regions or continents, put in the number of extinct genera, we can see that actually the largest numbers and the largest magnitude of extinction happens in geographic regions where humans have been arrived relatively late. Now, in Eurasia, prior to modern humans walking out of or migrating out of Africa and colonizing Eurasia, of course, we had Homo erectus and we had Neanderthals, and we may also have additional human groups, Denisovans, for example, present in eastern Siberia. So this means that, and of course, we originated in Africa. So these two geographic regions of Africa and Eurasia have had the presence of modern human or other humanoid population groups for much longer, where when humans entered Australia, North America, and South America, they would have met megafauna populations that were naive to human activity. So these are the 3 continental regions where there have not been humans prior to modern humans coming in, and these are also the 3 geographic regions where we see the highest magnitude of extinction. This has led to the natural conclusion that humans have had a large impact on causing these extinctions. So this is a picture of Paleolithic humans. Funnily enough, when you Google a Paleolithic human, they never wear trousers. So they had very warm legs and they only ever have a tire on the top. But here they are hunting a mammoth. But it should be said that it's not only direct hunting, it could also be indirect by land use changes for example, by burning. So it's not only through direct hunting, but also through other human activities. Now, originally, it was believed that this would have been rather a quick and dirty. So when humans move into a new region, that within 1,000 years, that megafauna populations would have been decimated, and this was known as the overkill theory. This isn't really believed anymore because as our dating, which we've also heard earlier, as our dating methodologies have become more accurate, and am I using the right word, precise or accurate? But as we've gained more precision in our dating, and also as more information has become available, it actually looks as if these extinctions were rather happened over thousands or tens of thousands of years, and not just within 1,000. So the human, otherwise had been called the human driver, the hypothesis is known as overkill. Then we have the climate, because it got warmer during the Holocene, has also amusingly been termed the overgrill. And then there've also been other drivers discussed, whether it was global pandemics, the over-ill, or the extraterrestrial impact that triggered the Younger Dryas around 11.9 thousand years ago, that made it glacially cold very quickly, whether this could also be a driver of species change and species loss. But really, it's only humans and climate that are being discussed now. And if I just go back to this, image here, it is super difficult in terrestrial ecosystems to tease apart human and climate as the main drivers. And that is because our human biogeography has been driven and shaped as much by climatic and environmental upheaval as those of the animals that we co-inhabit the planet with. So every time we have a major biogeographic push, it is closely linked to climate change, and therefore it is very difficult for us to tease apart whether when we have megafaunal turnover, is it due to humans alone, or is there also a climatic factor? For example, we pushed into North America at the major climate change between the Pleistocene and the Holocene, and the environmental changes that happened in association, such as the opening up of the Bering Land Bridge, which prior to the Holocene was which was a land bridge until it was closed during the Holocene. All right, so what I want to just show you is how we know all of this and what evidence do we have that, one, that species were even lost, but also what caused it. So some of the talks that we've heard today use different markers, but ultimately trying to understand the same things, which is what was present in the landscape and when did it disappear, and then ultimately trying to figure out why did it happen. So for late Quaternary extinctions, we use a multitude of data. Some we've heard about already, the fossil record, we have that also going back in time, but also, of course, now we have humans. So we use the archaeological record also. And the archaeological record is not skeletons lying in the landscape, more so it's charcoal that indicates an archaeological site and human use of a site at different points in time. Now, charcoal as well as bones can be radiocarbon dated to indicate when they were present in the landscape. So first of all, the faunal fossil record, as we've heard, tells us about what species were present. Also, importantly, about what species were present at the same time, which is not the same as what we see now. So as we go back in time, we're seeing these non-analog ecosystems, even 5,000 or 10,000 years ago, not very long ago, but where things were present in the landscape with other contemporary species that we don't see together today. And not only because of extinctions, but these are non-analog ecosystems as we go back in time. And then, of course, we can also estimate when a species is last found on the landscape, when it went extinct as a proxy for extinction. We've heard a little bit about the use of stable isotopes. So we use radiocarbon dating here. So this is the decay of the carbon-14 isotope, which, depending on how much of this isotope is present in a specimen, that we can use to quite accurately date at what point in time the individual died. So using this so ultimately we need a fossil chronology of individual species to understand when they went extinct. And we need to get that timing of extinction right in order to even start pinpointing the drivers. Now, this is a superb data set of mammoth and of Megaloceros, which is also known as giant elk. This is what we see up here, these massive antlers. And we have superb fossil records, which are not only temporally at very high resolution, but also spatially. And here this spans from Ireland in west to Wrangel Island in eastern Siberia. Mammoths are in blue and Megaloceros are in red. And here we see certainly a change in the presence of fossils across the landscape that can allow us to infer when they disappeared, but also why they disappeared. Now, fossils have inherent limitations in, we can't really say what's going on within a species. We can just say whether it's there or whether it's not there. And also there can be difficulties in figuring out two fossils, are they the same species? Or are they actually two different species, but based on the fossils that we have at hand, look the same? And that's why the advent of ancient DNA, so ancient biomolecules retrieved from subfossil specimens, has really opened a new field. And actually, paleogenomics was awarded with the Nobel Prize last week with Sven de Pebble from the Max Planck in Leipzig, who has done many of the methodologies that we also use for the faunal fossil record, but in humans, Neanderthals, and or paleo-humans, Neanderthals, and Denisovans, but that's a whole other talk. Anyway, we are now able to retrieve DNA out of these fossils that we can date to look at population level changes. So rather than just looking at species and when they disappear from a landscape, we can now start to understand the dynamics that precede extinction in order to understand why species went extinct and ultimately what are the drivers. Now, most of this data is from permafrost areas shown here in purple. So most of our data is from the Arctic area of the Northern Hemisphere. And this is just because the preservation of specimens is best for DNA when it's very cold, when they've been preserved cold. So fossils come out, and then we go into the lab, and then we extract DNA, but also other biomolecules, which I don't have time to go into, and stable isotopes being a super important ecological source of ecological information that we can couple up with genetics, just as Melanie mentioned in the previous talk, where we can use carbon and nitrogen to infer, for example, trophic level or what animals were eating at a given time. Now, based on retrieving ancient DNA from a lot of specimens within the same species across time, shown here. So we have time zero on the left. This is a seminal study on bison. We can then compare the genetic signature across time. It's like slices of a salami and looking how do populations change across time. And in bison, we can see that there's a boom and bust where there's a massive decline at the onset of the last glacial maximum, and this massive decline, this is all data from Beringia, happened way before humans arrived. So this is not a human-caused demographic change. Another more recent source of information to understand the presence of species across space is sediments, and to extract DNA from sediments to map out paleo communities of plants and animals, both mammals and vertebrates and other species. And this is a seminal, one of the first studies, or the first study using DNA from sediments. Here we have 4 different sediment cores with various dates. And what we can see is the presence or the composition of species is not the same. It's completely different when you go back 10,000 years to the last glacial maximum 20,000 years ago, and even further back. Species compositions change through time very quickly. And so this is an indication that the species compositions, as mentioned, that we have now have changed remarkably over very short timescales. Okay, I'm going over time, so I'm going to speed up with my last slide. Sorry, Dave, but I'm getting all excited about all the exciting stuff we can do with ancient DNA. I just want to show you here that this is a recent study which has taken samples from across the Arctic, and using sedimentary ancient DNA has then been able to, this is a very busy slide, I'm just going to show you the left side. We're going to focus here on the growth forms, but where we can get these temporal time series over the past 50,000 years of what species were present in the landscape and how that has changed remarkably. And this is all looking at plants, but coupling this to the megafauna extinctions, most of which, or many, are herbivores, and they've of course lived off these plants. We can start and tease apart why did they disappear and when. A last finding I want to show from this study is based on the fossil record. It has always been believed that mammoths disappeared from the landscape, mainland Eurasia at around 11,000 years ago, and only survived on Wrangel Island until 4,000 years ago. This is sedimentary. This is mammoth DNA retrieved from sediments. So no macrofossils. This is all sediments where you can't see anything except the DNA. As we walk through time, we see lots of mammals or mammoths. These are all the colors. Then we go to the early Holocene, mid-Holocene, by which time they should be extinct based on the fossil record in northern Eurasia. And wham, late Holocene, 3,900 years ago, there were mammoths. The last find we have actually is here. We have Wrangel Island, and just before here, that there were mammoths in mainland Europe or Eurasia only 3,900 years ago. And this completely changes our understanding of mammoth extinction, because based on the macrofossil record only, we thought they went extinct 6,000 years earlier. And this, of course, is important information for understanding the drivers. All right, flip, 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 flip. Basically, our understanding of late Pleistocene extinctions keeps changing. Every time we get new data, we have to revise our thinking. There's a huge disparity in the amount of data from different regions on Earth, and therefore we have to also keep revising. From South America alone, there are only 140 radiocarbon dates across 15 genera of megafauna. So to understand when something went extinct based on just one date is impossible. So we need to get additional data to start understanding for that area what were the drivers. But bottom line, new data constantly, we have to keep revising what we know. Thank you.
David, you're muted.
I should say det var bara flott, så tusen tack för det. We're going to move very brilliant stuff. So we're going to move on very quickly to Danny Schmidt, who's going to talk a bit about what paleontology has to offer in our understanding of what's going on today. So over to Danny now.
I hope this is in the right presentation mode. Yeah, thank you very much. So my contribution here is to take us into the future. I had the privilege of being one of the coordinating lead authors of the Working Group 2 IPCC report. And in there, we were tasked for the first time in the Working Group 2, which looks at impacts and adaptation, to have paleo in every region possible. And I have to say, in many of those, we failed. And so it's also a quest for us as a community to ensure that we are providing the information which is needed for those types of reports. What the report has very clearly shown and stated is that global warming has caused dangerous and widespread disruptions in nature. This is a step forward because what we traditionally would have had high confidence in is that we have had human interference of the climate system. Taking that forward into its impacts was new. And we're very proud that that was our first headline statement and accepted by all the 196 participating nations with whom we discussed this. And climate change is affecting lives of billions of people, despite our efforts to adapt. We have seen the floods in Pakistan recently. We have seen strong winds all across the Eastern American shoreboard. We've seen droughts in China, in Central Europe. We have seen the teleconnections of all of those, and we are currently witnessing extreme flooding in Australia. And so it's clear that climate change is not the future. Climate change is here today. What is quite important, and Elaine has pointed that out very clearly, is how stable our climate has been over the last millennia. So yes, if you go back into the history, and you can see here the records Elaine has just talked about, you can see the historical, the industrial revolution. You can see that overall our climate, during which we had the worldwide expansion of modern humans, was facilitated by this stable climate. It's definitely, at least since the advent of agriculture. The projections for climate change are just that, they're projections, they're our choice. What you can see here on the left-hand side, in those cryptic abbreviations of the IPCC, are combinations of economic, population growth, and climate CO2 expansion. And what you can see here is that under some of those, our warming would stop. Under others of those, it will take us significantly out of the envelope we will have experienced over the last 3 million years. And that causes a risk to nature's crucial services to us. We've heard a lot from people about the loss of pollinators, and that's pollinators from bumblebees and bees and insects and birds. We know of the importance of nature for protecting our coastlines, keeping our sediments in places. Many of us enjoy the recreation and just walking out in nature. But for others, the ocean, for example, or land provide incredibly important food sources. There are things we can calculate much less well. How do you value that every breath of oxygen you take is a breath from nature? So we have water filtration, we have clean air, we have climate regulation, all of which are under threat because of our impacts on the natural world. It's important, of course, to consider that climate change is not acting by itself. This is an unsustainable use of natural resources, so an overexploitation of our planet goes hand in hand with habitat destruction, with growing urbanization and growing inequity. And so it's really important to see those things together. Yes, we have climate change which has impacts on ecosystems and impacts on people. The way we react to those, the way we adapt to those, again, will have impacts on nature. But on the other hand, nature can help us to provide livelihoods and ecosystem services, but it also can help us to reduce the impacts on people. When we are looking at the geological record, and you saw some beautiful examples of what we know, the issue is that we usually have a better understanding from first principles of what happens to the drivers of environmental changes than on the ecosystem responses. So with this example, we know that fossil fuel and land use changes increase the CO2 in the atmosphere, that ocean and atmosphere are in equilibrium with each other in large parts of the world, and that therefore we have a lowering of pH. We also know that indirectly the warming is causing stratification, and increases in microbial activity, cause oxygen loss. But then when we take that forward to understand directly, causally, how it changes growth, development, reproduction, physiology, it is much more challenging. It is already challenging today in experimental setups. It is really difficult because every species does something different. Different developmental stages do different things in terms of responses. But of course, we are asked by policymakers to come up with assumptions on the migration of groups of organisms, the changes to ecosystems, the losses of biodiversity, the potential for extinction. And so upscaling from ecosystem experiments by themselves will not be able to allow us to do that. Those experiments are not long enough. They help us to understand the underlying physiology, but the upscaling is not their strength. Our records are messy. Our records are gappy. But what they bring is what Mike has also pointed out, the ability to have a look at a wide range of different events. You can see here on your right-hand side a different representation of geological time, starting here in the Permian, going into the Neogene. What you can also see is estimates of temperature anomalies associated with what we, as Mike has introduced, consider as the major extinction events. And these have resulted in changes in vegetation, in range shifts, in reef crisis and extinction. But sometimes for those, we are also very quick to deem causality. Temperature is what we can measure best. It's not necessarily the cause. And so when we, for example, talk about ocean acidification, nature has an amazing ability to buffer the system in itself. And if changes aren't really, really rapid, weathering on land and dissolution of carbonates on our shelves and in the ocean can buffer some of this. What the IPCC has concluded in our 6th assessment report is that biodiversity loss and degradation, damages to and transformation of ecosystems are already key risks for every region on this planet due to past global warming and will continue to increment with every increment of warming. We don't give very high confidence lightly. And so we have a wide range of evidence to which the geological record has contributed. We've also concluded in our technical summary, these are a little bit longer because we always have to have a traceability of what we do. The climate-induced extinction, including mass extinction, are common in the paleo record, underlying the potential of climate change to have catastrophic impacts on species and ecosystems. So what are prime examples of how we could contribute more to those analyses, that they go from very high-level statements like these to practical regional examples in our Asia chapter, in our Africa chapter, and in those regions where we're so desperately looking for them. One of the things we can do, and this is taking further what Melanie has talked about, is not just to look at what has caused the extinction, but to assess how long it has taken to recover, because it gives us an indication of the importance of some of those events. Extinctions also do not affect every species the same way, but we all tend to focus on one of our groups of organisms. And if we do more than one organism, then we're doing it usually in a small region. So what we need is to come together in fora like these, where we are considering an ecosystem-wide response, to see resilience as well and to assess those. We need good high-resolution environmental data as well as climatic models to causally link between biodiversity loss and environmental change. Sometimes it makes a good narrative, but we have no proof for it. And we need to be quantitative so that we can compete with the needs of the data quality. And we tend to like our mass extinctions. I had to say this in this forum, but we tend to focus on when things go horribly pear-shaped. It is also important to talk about resilience and resistance and assess where this has happened. Where do we might have climate refugia in the future? Where are organisms which, might not be as beautiful, but provide the same ecosystem services, so that we can think about how we can help with assisted migration, for example. And we have some of those time intervals, and I spent a fair amount of my career looking at one of those with the PETM, the Paleocene-Eocene thematic maximum. Its ages change from time to time, so sometimes it's 55.8, sometimes it's currently 56. What you can see here is the beauty of it is an amazingly easily recognizable event, because you can put your finger on it. So this is the carbonate records. This is the older rock here. It gets younger that way. This is Ellen Thomas, who first identified it in the marine fossil records. And you can see the white rocks turning into brown, which is the change in the carbonate compensation depth at this location over more than a kilometer, which is madness for anyone who's a geologist. And you can see then how it starts recovering and starts whiter again. Interestingly enough, more white afterwards. So there's an overshoot in carbonate production. The interesting thing is that this is an event which allows us to look at a system. We have marine cores, as you can see here, drilled with the joyous resolution, but we also have beautiful vegetation changes on land, beautifully mapped out. We can compare different groups of calcifying organisms. It's also time interval, which is really important as the base of the ungulates. And so we can think really of what it means for a system. What we need to be really difficult, what we need to be fully aware of, though, of course, is the time intervals. We are currently looking at a warming, depending on which decisions we take, over 100 years in the order of, let's be honest, 2 to possibly 4 degrees. Yes, we are all still talking about 1.5 degrees, but that's a dramatic behavioral change we would have in the next year or two. While here, what you can see at the PETM is a time interval of a roughly 100,000 year duration, but an onset which has been dated in terrestrial sequences who have much higher resolution of definitely more than 1,000 years. So we are an order of magnitude out in rate of change. And of course, rate is fundamentally important for organismal ability to adapt. But we see mechanisms which we see today. We have pole-wide expansion of plankton, which we can see now and which we could see then. We have assemblage, composition and shifts, which beautiful records of the continuous plankton recorder, 60-year record of plankton changes, is showing us the same today. The PETM showed short-lived organism coming up. It showed migrations of mammals on land, migration of the plant systems. It showed what we would call today red tides, dinoflagellate blooms, which we know when they appear today, wipe out fish stocks and shellfish and economies. We had extinction amongst of the benthic process, but not amongst some of the arthropods records we have there. And we have very highly resolved differences in species-specific responses to calcification. Everyone assumes this is a massive change in carbonate compensation depth. It's an ocean acidification event. Everyone assumes that this is negatively impacting calcification. And the geological record shows the complexity. Yes, in the laboratory, you take a foraminifera, you reduce the carbonate ion, it will be thinner. What the PATM shows, and this is to scale, a dramatic reduction in size of these benthic foraminifera. But what we have done is akin to what Melanie showed, is to use the power of CT scanning, looking here into the three-dimensional structure. So these foraminifera grow by adding chamber and restore they keep their developmental trajectory. And so we can do, hundreds and hundreds of specimens, something, no offense, Mike, someone working on dinosaurs would never have developmental trajectories of the same specimen at the same location. And what you can see here is the change in calcification of one species, Natalitidis trimpii. In the Southern Ocean, this is the Indian sector of the Southern Ocean, this is the Pacific sector in the middle, and then the Central Pacific here, which is a very oligotroph area today and then. And in the Southern Ocean, in a relatively shallow site, there is statistically no negative impact on calcification, but it's a very shallow site, there's a lot of food. Calcification is an energy demanding process, but this suggests if there's sufficient food available, the organism might buffer it. And that is something which we can also see in modern bivalves, for example. In other locations, we can see the expected reduction and then recovery we had anticipated. And so what the geological record can contribute is acclimation, adaptation, and regional responses. What I'd like to end on is to show that, of course, the geological record has various time intervals. What you can see here is the most beautiful use of the archives of the British Antarctic Survey at the Natural History Museum, using historical collections. So this is Scott's expedition to the Antarctic, that's Discovery. And it shows here the growth of Bryce-Owen's work by Dave Barnes and others, where it shows very stable growth patterns which change dramatically with the warming in Antarctica and the loss of sea ice. We often use growth in bivalves to study temperature. What if we turn this around and at a given temperature can have a look at the calcification response of those skeletal histories? And what we can do is then use a wide range of different methodologies and bring those together to think about what our understanding of data using methodologies where we are trained as geologists to work with very small amounts of sample, to use the CT scanning here in this example on coralline algae or marl, a very important temperate habitat former, using CT scanning, generating computer models and assessing how differences in growth structures, externally, these all look the same. Internally, they are very different and therefore very differently vulnerable to any kind of wave activity. We can map them out and link them to environmental protection in our protected areas. We can map them using similar methodologies Melanie talked about, in this case, scanning the magnesium concentration under acidification, and then use the whole narrative to put it together to generate engineering type finite element models to understand if those organisms are more likely to break, if we have more fractured material, and therefore if we're losing habitats. Because ultimately, the question we want to address is, where shall we protect? Because we can't protect everywhere. How can we ensure that we have diverse habitats in those areas we're protecting? How can we network marine and terrestrial zones where migration is possible for land mammals, much more difficult for plants, as Shengju talked about, because they're so much more dependent on their soil, where we could zone around it so that when organisms are on the move, that where they're going to be in 20 years' time will still be protected, and identify refugia where we can support assisted migration. Because ultimately, we have to act, and we have to act now.
Well, thank you. Thank you so much, Danny. You brought such a very strong paleontological focus onto the huge challenges that we face today and that we have to deal with immediately. So thank you so much. We're going to move on now, and we're going to look at some big data, and we're going to look at modeling, and we're going to look at the whole rock record. And Jun Swan is going to present some of the artificial intelligence algorithms that he's been working on with really big data sets. So over to you, Jun Swan.
Yeah, thank you, Dave. And I want to say thank to Mike and Dave to invite me to attend this important activity and give the talk. So I will introduce our recent work. We try to use AI algorithms and a supercomputer to help us to search the global 3D graphic records for Earth's hidden extinction. So in 2005, the Science magazine raised 125 questions. What don't we know? So there are several of them related to the biology. So the biodiversity, for example, what determines species diversity, and how and where did life on Earth arise? So if we think deeply, so we can find some more difficult questions. For example, how many species on the deep time Earth, and how they get evolved through the geological time. So we can come back to three major scientific questions. First, how did the present complicated ecosystem form? And how did life evolve from one form to another? And was there any extinction or biodiversification event happened in the deep time biosphere? How can we recognize them? So if we want to answer those questions, first we need to understand the deep time biodiversity history. So to answer these questions, there are several important work has been published in the past 30, 40 years. And one of the most important ones was conducted by Jack Sapkowski in 1981. So he organized a personal database of paleobiology, of paleontology. So the database is very simple and hosted on his own computer. So it can included only 3 fields. So the fossil names and the first appearance and the last appearance. But such a simple database can help us to understand, to give us a quite direct understanding about the life history in the Paleozoic. So to the left, to the right, you can see the biodiversity curve. So you can see that here shows the Paleozoic diversity pattern at the family level, and also he generated a genus level too. So it's based on 2,800 families, and the time duration is about 650 million years. And we can see that there are several major biodiversity values in the curve. So number 1, 2, 3, 4, 5, and which was named as a Big 5 extinction. And we can also see there are 2 major radiations in the Earth's history. So one is in the Cambrian to the Ordovician, and another one in the Mesozoic to the Cenozoic. So so we can see that the temporal revolution. So he divided the whole time duration into 82 time beings. So that means the average temporal revolution is about 8.1 million years. So after about 20 years, there come out another famous work. So another database, online database, which called the Paleobiology database, was constructed by a team from John Arroyo and also other colleagues in the United States and Europe. So we can see that the database is very complicated. So it contains over hundreds of fields in the database, including taxonomic, paleogeographic, paleoecological informations of each fossil occurrence. And through such a much larger databases and the big data from that database, so they published a new biodiversity curve in 2008 in Science magazine. So they use about 300,000 fossil occurrence from the global database, and the average temporal revolution of the time being they used is about 10 million years. So we can see that the curve also shows some radiation and also some biodiversity declines. So if we put the two curves together, and we will be able to see that there are some similarities. For example, some of the mass extinction events, but there is also some major difference between the two curves. So which one is real, and which one we should rely? So to answer those questions, we think that we need a high resolution biodiversity curve, because we think that because of the two curves, they have different temporal resolution, and the temporal resolution is quite coarse. So we can't recognize the details of those events, especially for the extinction event. Most of the extinction events, they happen very fast. So if the temporal resolution is only, for example, 8 million or even 10 million, we will not be able to recognize those rapid change in life. So there is one method designed by Peter Sadler in 1995, and he designed a program which called CONUP. And in 2009, they published a paper in JSA Bulletin, which called the High Resolution Early Paleozoic Time Scale. So they use such a method, quantitative stratigraphy method, and based on a global database of graptonite, only one fossil clade, and they reconstructed a much finer temporal resolution biodiversity curve, but it's only for the graptonite clade. So we can use such a method to help us to create a high resolution biodiversity curve. But remember what I said, they are only focused on one fossil group. So that's graptonite. So they only use about 20,000 species. Now we have about 10 million species on Earth. And the number of species in the Earth's history is much bigger than that. So we will need the big data. So if we have a much larger big data set, we will be able to make some more reliable analysis and prediction. And then we also need some new algorithm to help us to establish the geological time scale. And also because it's such a large data set, and we will need much larger computing power. So we will need to use a supercomputer to help us to do the data analysis. So first, where does data come from? So we designed two databases and use them to collect the data. So one is called the Geobiodiversity Database. We call it GBDB. It's also the formal database of the International Commission on Stratigraphy. And one stratigraphy is another database we designed recently, which can be used for the data sharing and between databases. So through those two databases, we could be able to save the stratigraphic legacy data, which has been preserved on papers and also stratigraphic report from the museum, from a library, from a geological service, or scientists' personal shelf or computers. And we have a stable data compiling team, and we hired about 6 ladies working for the project for nearly 5 years. So they just extract the information from those literatures and input them into the online database and make it open to the public immediately. So after 5 years, we have the data available for this project analysis. And here shows the result. So in this study, we focus on the data from China. So we digitized about 80 to 85% of all the published sections in China. So it contains about 10,000 sections and 500,000 fossil occurrences. And so we can see that the data in density covering the whole China, even in Tibet and the terrain. And also, if we go back, we look at the paleogeographic distribution of those data, and we can see that even they come from one single country, the present country, but actually in the Paleozoic, they cover quite a wide range of geographic range in the lower and the middle latitude from the Laurentian. So the second technical question problem we need to fix is about the geological time scale. And the left figure shows the international standard published by the International Commission on Stratigraphy. It's called the International Coronal Stratigraphic Chart. And for example, for the Paleozoic, nearly 600 million years, and it contains about 2 GSSPs. So that's the golden spikes. So that means the average temporal resolution is about 5 million to 6 million. So we can use such a standard geological time scale, but we can get what we can get. We will get a similar curve like this one. So it's obviously not the time scale we want to use. So we need to create our own. So we can come back to the basics. Why are time scales difficult to build? So there are major 2 factors. One is that different locality or sections they may present.
Okay, the thing.
If we want to.
Oh, right, you're back. You were breaking up a bit, Jun Xuan. Yes, I guess it's the thing we fear most is the break in transmission.
Oh, yeah. Can you hear me now?
So we can see you, but we've lost your screen.
Okay, I will share my screen again, but you can hear me, right?
You can see and hear you. You're fine.
Okay, sorry.
Perfect.
Okay. Yeah, so the observed 3 graph sections connected with each other in detail concerning the sequence of the first appearance and last appearance event of fossil taxa. So that's the reason. So we need to think like a computer. So just think about 2 sections with 2 fossils. So the section A and the section F, they both have the green fossil, the red fossil, and the green fossil. So in section A, they occur together, and in section F, so the green appeared before the red one. So there will be about 6 different possible sequence of the first appearance and the last appearance. And the sequence 1 and 6, they are unacceptable because they didn't include the observed coexistence. So we have the sequence number 2 to 5. And so we check those 4 solutions, and we will be able to find that the sequence number 4, this one, will be the best one because it requires a least net adjustment of the observed regions if we want to make the 2 sequences consistent. So if we extend the first appearance of the red one to the same level of the green one, then the 2 sequences in the 2 sections will be consistent. So we call it as a range extension. So we can use that to find the best fit sequence. So then we can just think like a computer. But the problem is that if we have only 2 species, 2 taxa, the possible sequence is only 6. But when we have 7 species, the possible sequence will be about 600 million. So it's a big number. But for 100 species, and the number will be 10 to the 300. So it's a huge number, and we can't make it by ourselves. So we need to use a computer to do that. And so there is a program designed by Peter Sadler, as I mentioned before. So it's called a counter program. So use that program, we will be able to. Sorry. So use that, we will be able to do the computing, but this method can provide a quite high temporal resolution, and the resolution can increase from the traditional 8 to 10 million years to only 30,000 years. But it requires a very large computing power. For example, for 10,000 species, one calculation will require about 17 years. So it's impossible for our scientists to make the calculation. So we need the supercomputer to do that. So we started the programming since 2013. So first we visit Peter Seller at Riverside, United States, and we designed the first CONUP program, which called CONUP.net, and it's about 3 times faster than the original program designed by Peter. And then we work with another team. We designed another program which called CONUP.HANA, and it's even faster, but it's still not enough. So in 2015, I decided to start our supercomputer plan. And so our team, we don't have any experience in the supercomputer programming. So we just started from the beginning. And we spent about 1 year to learn how to do the computing on the supercomputer. And we get the first parallel computing version in 2016. And 1 year later, we get a very good version, and which can be about 500 faster than the original version. And we can use about 10,000 CPU cores on the supercomputer in China. So here shows the results. So this is a curve we got from the supercomputer. So it's based on 300 published stratigraphic sections, and it contains about 11,000 species, the marine invertebrate species. And we can get such a detailed biodiversity history for the whole Paleozoic. And we can recognize the several major extinction events over there, the End-Ordovician, late Devonian, and the End-Permian mass extinction. And it can give us more details about fluctuation, about diversity through time. And also we can compare it with the previous study, for example, John Arroyo in 2008. And here shows the blue curve is their curve. So you can see that they have only 25 data points, and we have 11,000 data points. So the resolution increased from 10 million years to 26,000 years. So it's about 400 times better than the previous one, and it can give us much more details. And also we can see that there is something different in the two curves. For example, our curve shows a diversity loss of valley here, but their curve shows a different pattern over there. So that's quite interesting questions we can answer in the future. And also here shows the different clays presented quite different patterns in the Paleozoic. For example, the trilobite, graptolite, conodonts, cephalopod, all those major groups, they have different patterns through time. And even for those major extinction and radiations, they have different trajectories at that time. And also we try to find out the problem about the temporal resolution. So we just reduced our temporal resolution from the 26,000 year to 1 million year to 2 million year, 5 million year, and 10 million year finally. And also we go to the PBDB time being, and we can see when we decrease the temporal resolution, the curve changes their shape. And at least we need to use, for example, this time being. So this light blue one, it can still keep the original information about the biodiversity change. Otherwise, if we use a coarse temporal resolution, I mean, some of the details were lost in the biodiversity curve. So that means the temporal resolution really matters in our biodiversity analysis. And also we try to correlate the marine biodiversity and the environmental proxies that are available. And one thing I don't want to mention is that when we publish these papers, there is no high resolution environmental proxy data available for the Paleozoic. So we need to reduce the resolution of our biodiversity curve to the same resolution of those environmental factors, and then we can make the correlation. And we found that there is no relationship between the biodiversity history and those environmental factors except one, so that's CO2. And we found that there is a quite good correlation between the biodiversity and the CO2 in the Paleozoic. And also we can find that the correlation decreased at the early coupling first. And we think that probably related to the decrease of the CO2 in the late Devonian to the early coupling first. But we need to find more evidence to prove that. So this research work showing the power if we can link the geoscience, data science, and the computer science. And after we publish this paper, there is some report on this article. For example, machine learning, it reduces uncertainty in understanding the Paleozoic biodiversity, and the supercomputer help us to score the fossil record for the Earth's hidden extinction event. And after this work, it gave us the evidence, the confidence that we can make some more detailed work through the Earth's history. So there are many mysteries in the life history in the deep time Earth. And in the next 5 to 10 years, we will focus on those major extinction event and radiation event, and try to change our understanding about those major biological and geological events. And one thing we want to do is that we will try to use AI in the data collection and data analysis in the future work, and also the data mining. So I can show you one example. For example, we know there is a knowledge exploration in the past 20 years, and this figure shows the exponential growth in the number of academic publications. And how can we read all those publications by human? The answer is no. For example, for a PhD student, he may be able to read 1 publication in 1 day. And for an experienced professor, maybe 10 papers 1 day. But for a machine, if we train the machine in a better way, the machine will be able to read about 5 million reference in 1 day. So we can see the potential if we can use the AI in the data extraction from the literature and help us to find the new knowledge from literature. So we are still working on this part, but maybe we will be able to see some preliminary result from that in the next couple of years. So thank you very much. That's my talk. Dave.
Yeah, okay, thank you, Junchuan. Absolutely incredible work, and moving away from certainly when I was at university, the sort of anecdotal side of science that we pursued to real strong data that we can now efficiently use to test our various hypotheses on the deep time stratigraphic record. So thank you so much for that. So a couple of things now as we move towards the panel discussion. And as I thought, a lot of the talks have run over time, but they've been extremely valuable. And I think it was really good that everybody had time to develop their arguments properly. The first thing I should say is that there is a poll, which I'm going to launch now. And there are 5 questions on this poll that members of the panel came up with. And they're very interesting questions. So we're going to ask you to vote on each of these questions. And I'm launching the poll now. And I think you should be able to do something with that. I'd like to thank all the speakers. The talks have been absolutely marvelous today. And I think everybody has learned a lot from these very diverse talks using all sorts of different evidence to tackle a variety of different problems, and it's been quite wonderful. So with great joy, I hand over the chair to Professor Mike Benton, who's going to move on to the next part of the proceedings, the panel discussion. So thank you very much, everybody, and over to you, Mike.
Thank you very much, David. And we've been collecting questions from the panelists. We also have some from listeners. So if you're attending this, please submit any questions you may have into the Q&A button at the bottom of the page, and we'll try and pick those up as we go. I'll start with a few questions that have come from the panelists, and we'll see what answers we get. These are important issues that are affecting us for the future. And so a question from Melanie, which I don't know who could tackle this, is what can paleontologists and geologists do to help the IPCC? Unfortunately, Danny is no longer here, but we've had some hints in her talk. And I don't know if anybody would like to suggest what more can we offer from deep time research to the IPCC in helping them make recommendations.
I can elaborate on the question
Thank you,
a little
Melanie,
bit. So the thing is that I had never thought of this until she mentioned it. And then I thought, why have I never thought of it? I go to conferences, I attend meetings, but I've never been in touch with anyone who said, oh, yes, please provide your data to the IPCC. And what she showed was that there's this real big hiatus, apparently, in what's coming from paleontologists. So maybe I need to contact her and ask her what I can do. But I think step one would be to have maybe a bigger presence of the IPCC at conferences, or at least some link there, because so far, most of the people who work in paleontology, there's some evolutionary biologists that sort of dive into the deep time. And I know that there's biologists who've never had geology, similar to Eileen, who dive into paleontology, but there should be the reverse as well. And I'm missing that. And I think that we need to collaborate more, and then we can also find out what we can do.
Thank you, Melanie. And I wonder, Elena, can I throw this at you? Because obviously you've talked about Pleistocene extinctions that were perhaps caused by, to some extent, by human activity. But how can we frame, or how do you think we can frame deep time questions, what we learn, to help the modern conservation movement?
I mean, it gets difficult when you go far back because you have that uncertainty of time. And when we're working with years and decades and ultimately no more than a century of massive scale moving forward, that we also need to have an understanding at that time scale in the past. So, and I think only really the methods available for us to understand the last 20,000 years and how environments and ecosystems have changed is perhaps a more reliable time frame for direct sort of utilization in the current the current global challenges. And I think there are in IPCC there are scholars who are aware of the research, but I think just as when I moved from ancient DNA and started incorporating stable isotopes as well, which to me was hugely important to understand any genomic change, you need to understand the paleoecology too. These are two crowds that go to different conferences and don't mix at all. So as with all of us, it's like we have our areas of interest, and then we go to our echo chambers where we all kind of do the same thing, and we might disagree, but we sort of have the same disciplines. And I think of this of having much more interdisciplinary meetings like the INQA or the quaternary meeting, where rather than having sessions which are very siloed, to have these much more sort of interdisciplinary sessions so that we can learn from each other's and the methods used. Because I think the talks that I've heard today have been relevant and fascinating, and I'm sorry I haven't heard, well, some of this is so new, but I'm sorry I never go to these kind of conferences to hear this kind of work, and it's hugely important for my work. And likewise, also moving into the future. But I think really this time frame of, and we are talking only decades, I mean, by 2050, we'll have a sea ice-free summit in the Arctic. This is within our lifetimes. So we need to perhaps look at the shorter timescales of millennia at most, and use that information directly to informing our conservation measures moving forward. So in that respect, 500 million years ago is more difficult.
I'm sure you're right, Elena, that that's why I asked you, of course, because you're dealing in hundreds or thousands of years and dealing with taxa that have close living relatives, and you get DNA, for goodness sake. That's very helpful. Not mentioned so far, though, has been that there is a discipline called conservation paleobiology, and some of that is enormously informative. I just hope people will listen. We haven't really represented that here today. And just to give one example, Catalina Pimiento, who works on modern and fossil sharks, did a beautiful study a few years ago where she crossed the boundary from Neogene into the present day, made an ecological argument that shark genera and species are quite long-lived, therefore you can actually look at them over hundreds of thousands or millions of years. And because they are top predators, the health, and that therefore they live in small population sizes, therefore the health of the shark populations in the oceans is quite a good barometer of the health of the whole ecosystem. That's quite a big claim, but if you accept that, then you can look at the evolution of a group like sharks, where there's quite a good fossil record, and cross over between past and present and try to make some predictions about it. So I don't know if there's any other comment on that question. This is about the what can we learn. Yes, thank you, Jung-Xiang.
Yeah, so actually I want to talk about the temporal evolution. So it has two aspects. One is that, I mean, the present paleobiology or ecology studies, they focus on the change from days, weeks, months, and I think I'm probably to the 100 years. And I guess the upper lift is about 1,000 years. But our geological study, we focus on studies changes many years.
Yeah.
So first thing that our geology or paleobiology study, we need to try to reach the final temporal evolution. For example, like tens of thousands of years or even 1,000 years, so that our result can be compiled or taken into consideration by the present paleobiology or ecologist. So that's one thing, one aspect. Another aspect that the present study, we can only look at the present change. So generally it's only, for example, if we think about our cultural history, only hundreds of years. So we cannot precisely predict our future. For example, after many years, what will happen to the life on the Earth? But if you go back to the deep time, there are so many cases over there. They already happened over there. So if we know that, for example, if we cut off all the forests on the Earth, what will happen? We can go back to the Earth and find there are already some experiments over there, and we can learn from that. And we will not need to suffer such an extinction, and we can know what will happen. So I think there will be two aspects.
That's really helpful, thank you. Yes, the short time scales we can achieve, and we have run many horrible experiments in the history of the Earth. We don't have to do it again. I think David would like to comment.
Yeah, I just wanted to come in that we've certainly in the UK and probably Ireland, we've seen a fascinating series of programs by David Attenborough on the Arctic and the Antarctic. And I was going to come and pose this to Elena again, because I know you've done some fantastic work on polar bears. We talked a little bit about sharks as being sort of apex predators. And what are you seeing in the polar bear populations, and how is that working out?
We just submitted a paper this afternoon. It's on bioRxiv, but where we see, this is where we've looked at populations across, where we can model the last 11,000 years of population change around Greenland. And interestingly, we see this and this is using genomics and stable isotopes and geometric morphometrics and also habitat modeling. And we see this incredible association between changes in genomically derived population shifts. So where we see it, we see across 11,000 years, every time we see a major climatic shift in temperature. So we see an increase in sea surface temperature and a decline in sea ice. This happens 3 times over the last 11,000 years. At the same time, we see a genomic change in diversity, a dip. So there's this very tight association between shifts in climate. And here, and I think actually that was very good, that we're looking at temperature because this is the easiest for us to measure, but it might be something else. But we look at temperature and sea ice change. So we see this decline. We see habitat suitability being pushed up. So they're declining because they're the suitable habitat keeps being pushed further and further north. And we could then model into the future and see, okay, well, what basically, so we have paleoclimate data, really high resolution going past 11,000 years. And when we see this magnitude and rate of change that we expect over the next 100 years, how do we then expect polar bear populations to change? And that's really a sentinel species that is an apex predator of Arctic marine ecosystems that ultimately mirrors everything that goes on beneath. But just one comment is this, that I've talked about non-analogous communities, is that when we survey, and I mean, the more data becomes available, the more we can see sort of high resolution, is that each species has an idiosyncratic demographic history and an idiosyncratic response to the combined stresses of humans, climate and environmental change. So it is very difficult based on just one species, and that's why we need these multiple species approaches to understand or to predict what will happen over the next 100 years.
Thank you very much, Elina. And you've moved helpfully into another question which Jen Chu had asked, which is, are there indicator species or indicator regions on Earth today that we could use as an alarm call to say, this is terrible? And I guess the answer partly might be the polar bear, the Arctic. I don't know if people have other ideas, Elina, maybe.
Yeah, there's also, I mean, the issue, well, the positive thing for the Arctic is that because we have Arctic amplification of whatever goes on the rest of the globe, the magnitude is much higher. And we also expect then for ecosystems to become much more stressed. But when we go back in time, the Arctic has actually experienced major shifts, whereas we're in the tropics, they haven't. It's been relatively stable compared to the polar regions. So this means that moving forward, if we model back over 20,000 years, really there's been very stable environments across the center of the planet, or sort of across the equator. So the issues being moving forward, when there's massive change also around the equator, these ecosystems are they've not tried this before. So they're going to be naive relative to ecosystems in the Arctic that have gone through this over the last 20,000 years. So that spells disaster for the tropics, because there we have a lot of species that have very small ranges and they have nowhere to go to. So really looking forward with habitat modeling, by observing what's happened over the past 20,000 years and what are we going into, it looks as if the tropics are going to be the worst hit, because they simply don't have the resilience that Arctic ecosystems have, of having gone through these major changes over the last 20,000 years.
That's a very good point. And I think we see the bad effects in the tropics with, for example, the Sahara Desert increasing in size by many kilometers each year, and people fleeing because nothing can live. It's not just the people, it's the plants and animals, of course, cannot live anymore. Once the temperature at the equator goes above whatever, 30, 35, it becomes impossible. Any other comment on this question of indicator species? Yes, Melanie.
Yeah, so because we've by now lost already quite a few species in our lifetime, in our recent years, that have been, I think the black rhinoceros, that have been referred to like, oh no, it's going to happen, and then it did. And then everybody was like, and then we moved on to the next one. So forgive me for putting it bluntly, but I would actually say that looking at these indicator species might actually be a bit of a trap, because when it comes to this sense of urgency and this sense of we need to do something, and especially when it comes to those that can actually do something, because it's not something that we as normal citizens or even academics can solve. I would say that we need to focus maybe a little bit more on our own species, because if you look at the floods and the amount of people that died in Pakistan quite recently, this is not something that can keep happening.
Thank you. And Jen, you would like to comment?
Maybe Alina was the first one to raise her hand. I will go after.
No, I just wanted to also, this, Melanie, that I agree that, I mean, our own species, because we are, I mean, how how our agriculture has developed has been in such tight connection with the seasons and the patterns of rain over 10,000 years. And that is shifting so quickly that we're also putting our own existence at risk. And these ecosystem services, which is clean air and clean water, that we're really rather putting a spanner in the wheel. And I think we keep forgetting, we keep disconnecting the fact that we're also very much a part of nature and of the ecosystems. I mean, we are embedded and entrapped with these ecosystems. So once they come out of when they reach a tipping point, so do we.
Thank you. Yes, Jane.
Yes. When I was looking at the modern modeling stuff, I realized that the equations they used were based on observation along 10 years or 20 years or hundreds of years. But there are so many things in our system that will function only for thousands of years or millions of years, like the plant alternation. During the human come out, there was not a big influence of the vegetation change, but that influence will make a big impact on the system and also the weathering stuff. So we should really cooperate with the modern scientists to add more long-term circulation things into that. Because if I do see something died out, that will be too late. Because when the disaster want to happen, they will happen quite fast. But there will be a long time before the effect gathering and explode.
Thank you. I'll move to a question we had from one of the listeners. We can come back to the present day crisis, of course, but there was a question about why do we talk about the big 5 mass extinctions, and what about the Ediacara, and what about the late Cambrian? So I don't know if anybody has a comment on the big 5, and the current extinction is the 6th. I suppose maybe we're reluctant to say the big 7, because then the current event would be the 8th mass extinction, and then it would get very confusing. Or each edition of Colbert's book would have to be the 7th, the 8th, the 9th. Anyway, any comment from anybody? Maybe David could start, and I don't know if Cameron is there. We can also get an opinion there from Cameron. Why do we just talk about the big 5 when there were other big events?
Well, I think the Ediacara biota is a really interesting one to think about. I call it biota because there's still a lot of uncertainty about how many animals are actually within this biota. It's soft-bodied. The organisms are large. They're more complex than anything that's come before, and so they are a very distinctive entity. Dov Silaker called them aliens on Earth. He called them Vendozoans. He said they were quite distinct from Metazoan evolution. Now, in fact, we've swung the other way, in which people are starting to identify the roots of many animal groups within the Ediacara biota, and there's certainly skeletal organisms, little tube-like things. There's reef building organisms too within that time frame. But the dominant elements are the sort of frond-like organisms that I really don't have a strong opinion on this. I'm not an expert, but they dominate the Ediacara biota, and certainly. There is I think by any measures that we use for the 5 big extinctions, there is an actual certifiable extinction of the Ediacara biota at the end of the Proterozoic, and then we come into the Cambrian explosion and animals take off. But nevertheless, there are elements of that Ediacara biota that still persist for some time, and that happens in any extinction event. There are these taxa that continue on through the extinction event. We don't get 100%. Thankfully, we don't get the 100% removal of these animals. So yes, I would agree with the question. Perhaps we should be thinking about the Ediacara biota as a 6th extinction within the fossil record. And we may just ignore it because it's in the Precambrian and we are too much focused on the Phanerozoic and animals and plants rather than other organisms.
Thank you. And Cameron, welcome. I'm sorry we missed you earlier, but it's great to have you join us. And this question of the Big Five, of course, you were going to tell us about the late Devonian, and oddly enough, there were two quite distinct events in there. So why do we call it a single event? Or would you like to claim that both the Kälvasser and the Hangenberg are each independent mass extinction?
Well, hi everyone. Sorry I've jumped on here so late due to your call today. So I'm looking at Middle Devonian extinction events. And these are things as well that are, they've also like the end of the Ediacaran or the Ediacaran biota. They've sort of just been pushed aside and no one's really looked too much into them. But I think what one is seeing over here now is that, of course, extinction is really the norm. It happens, but I liken it when I think about it, the tree of life is the severity. Does it hurt the tree more to cut the leaves off, to cut the branches off, or to chop it right down at the trunk? It depends where, I guess, in that taxonomic rank in terms of severity. So, yeah, what we're seeing, we haven't really worked much or read much or done much work on the late Devonian stuff, but from what I've seen in the Middle Devonian, it was as severe as the late Devonian, depending on where you were. As Lena was saying earlier as well, but I just jumped in now, so this is fascinating. Someone else who works in high latitude environments and conditions is that at the high latitudes, things get amplified, and depending on where you are, that extinction event or those drivers for extinction can be more powerful than others. So organism might be able to cruise through the event, whereas others that live in very marginal conditions who are quite hyper specialized will really struggle. So that's about my 2 cents worth on that for now.
Thank you, and I'm sure we can all come up with additional events like the End Guadalupean, the Carnian Pluvial event, and there are many more that are somewhere in there. I think we just have to stick with the notion of a Big Five probably and accept there's a sliding scale. Yes, Melanie.
I just wanted to, I don't think any of us are actually ignoring the smaller extinctions. Some of them are actually quite significant. It's just that when you have to make a decision on which ones are, I would say, almost globally, and especially when it comes to like a massive difference in ecology after an extinction. But it's a fine line, and I think Sapkowski actually sort of made us choose the most disastrous ones, but that doesn't mean that the other ones weren't there or didn't have any effect or that we dismissed them.
Thank you. So I think we'll have a couple of questions on a theme looking to the future, and then maybe that will be enough. We'll see how things go, whether any more questions pop up. But in other words, what I want to, yes, and Elena, if you have to go, of course. And thank you so much for your contributions so far. The final question, or a further series of questions, are about what do we do next? So one thing that we do next is obviously we try to get the attention of the IPC and the modern conservation community to say, hey, we can get involved, there's certain stuff we can tell you about. But within the context, the specific context of our own studies, what are the future opportunities that we have in the sense of data and quality of methods and kinds of questions? Any thoughts from anybody about that? The next 10 years, the next 20 years. I'll make a brief start just to get the ball rolling. I started my interest in the end Permian mass extinction in the 1990s, and the best review that you could give was that the event may have spanned as much as 10 million years, much of what we would now call the middle and late Permian, with numerous bits of extinction scattered over time. And this was very poorly understood because of the quality of dating was very poor. The time bins were huge. There was a mixing of the end Guadalupean and the end Permian event as if it was a single event. And now we look, 15 years later, wow, how things have moved on. It's quality of dating, access to new sections in China, all kinds of reasons have moved us a great distance. Can we imagine what we can achieve in the next 10 or 15 years? Any thoughts about that? Jane, your hand is up, but I'm not sure if this is a legacy hand or if it is a new hand.
My hand was always up, I think.
I'll ask Jung Xuan, because you were talking about certain, you can maybe summarize from your own talk, in fact, what you would say are some of the possibilities.
Yeah, so I'm thinking about several possibilities. One is that when the geological and the paleontological data increased, and we have the because there are several big scientific programs in geology, like Deep Time Digital Earth and also the Earth Cube. So I mean, they try to establish a platform for the communication between geoscientists and the computer scientists. So we can talk with each other. We can tell them about our requirement, so they can help us to fix our technical problem. So I think in the future, we don't, I mean, some of the studies, they will go much further. For example, we will not use, for example, only the papers that we can remember in our mind to create a pattern, but we will be able to use a AI program or some big data analysis technique to help us to search all the reference and get the data from them and put them together and create the pattern. So there are already some case studies by Shannon Peters and other teams recently. For example, one paper last year on Science Advance is talking about the pyrite evolution. So it's a very good example. And also recently there is another paper on Nature Communications just last week, talking about some deposition change through time. So I think such kind of literature papers will come out soon by soon. So we will be able to say 10 or 20 next year. So I think probably in the next decades, 10 years or 20 years, we will be able to see such a change in the research paradigm. So people, they will go, I mean, at least some people, they will go further with a computer data scientist, and they will use the data science technique to help us do the research work. So I think that's one thing that I want to mention. Secondly, is that just as Mike mentioned, so about 20 years ago, when I started my research work, we generally go to the field, we collect sections. The section is about, for example, hundreds of meters thick. We'll go there and make 10 sections, 10 collections, 20 collections. But now we go there, we collect the sample centimeters by centimeters, because we want to get some more details. We want to reach from many years to maybe tens of thousands of years. We try to get more precise data from the field, and then we can reach the temporal resolution of the modern ecological system change. So I think that would be another important direction. So that means the people, they can use our data, our analysis to predict our future. Yeah.
Fantastic. Thank you, Jung Xuan. And I think David had a comment.
Yeah, it's just following up on some of that, and certainly within IUGS circles, it has been said that maybe geologists concentrate too much on the past. Well, that's what we actually do. But I remember that Chris Scottese a number of years ago did a lot of forward modeling of his maps, plate tectonics and paleogeographies, and looking to the future. And I'm just wondering about in this big science program that you can develop quite a number of different scenarios with these massive databases that you can actually test moving forward. And I wonder if that's part of the program, or is it just too frightening to actually do?
Thank you. Any other comment? we'll wrap up in a minute, I think. So I don't know if Cameron, if you have any thoughts about, in general terms, where you think we can go in terms of step change or new topics.
Well, I think as was mentioned earlier, is that of course with the more data we collect, the better our models become as well. I guess as well we must also be careful that we don't zoom too much on error. So a lot of what we do, it's what I always tell my students that I lecture and my staff as well, is that in geology we work with error margins. Everything we have has got large errors on it because we of course are observing it hundreds of thousands or millions of years down the line. So whatever we zoom in on, we must make sure that we're not zooming in on error. And one of these ways, of course, is to add more data to it. So there are probably even methods and procedures and what have you that we haven't even thought about yet that are still on the horizon. And I think that us as geologists, I think we're big dreamers. We're an intrepid bunch. We're very romantic in a lot of ways. And I think we should continue on in it, stay, it's okay to be odd and to love these things. And I think that will come in time. And I'm actually glad that there are error margins, because then there would be nothing to talk about. There'd be no hypotheses to throw around, no one to fight with or argue with at conferences, etc. So I think it's a wonderful job. I wouldn't do anything else. And I'm sure that there's a lot more in future to come.
Fantastic.
So I'll leave on that part.
That's brilliant. Thank you. And I think we have to have a final word from Jen and Melanie, who are both doing their PhDs. So the future is with you two, as well as a little bit with us. But very good to hear if you have thoughts about where the subject may be going. I don't know if Jane, you would like to reflect on what may be possible in the future.
Yes, I mean, there are so many extinction events that were caused by a sudden disaster, like the other things, or the volcanism eruption, which we cannot predict. So what we can do is read all the history and get all the possibilities clear. For example, what if future becomes a super greenhouse? What can we do? If the future becomes frozen again, what can we do? So what we can do as a geologist is to study as much as case study as we can. So I have only done the Permian-Triassic, the super greenhouse condition for the vegetation. I think the next step, I will do more like Carboniferous Permian on the glacier period. So we can see, we have more backup plan B.
Fantastic, thank you very much. And Melanie, can I ask you for a reflection on where we're heading in our research?
So I've jumped through time so far in my career. I actually started with the early middle Triassic recovery phase of the Permian mass extinction for my bachelor and other undergraduate research projects. And now I've actually, after the end Cretaceous extinction, dived back into the Devonian, which also has a very odd mass extinction, which sadly couldn't really be attended to today. But I like zooming in on extreme small details with multiple different techniques and used by different disciplines to sort of shed light on the smaller circumstances, the less geological circumstances, if I may. So I like that's, for instance, why I timed the season of the impact, because I think these smaller things, they matter, and they especially matter when it comes to relatability. Just imagine a sudden disaster happening today. If it would happen in spring, the consequences are far different than if they were to happen in winter or autumn. And I like trying to figure out these elements. So I want to look at mass death deposits, and then first of all confirming if everything did indeed die simultaneously. And then once I can confirm that, then I will try to peel away, get closer to, so what happened to them locally there? Because I think that zooming in, that's becoming possible nowadays. And that's something that wasn't possible 10, 20, 30 years ago. And it's something that I'm incredibly hooked on, and it keeps me up at night. So that's where I'm going.
Fantastic. I think that's a great way to finish the discussion. Thank you so much, everybody. And I'll hand back to David, who I think may report on the poll of attenders.
Yeah, I've the very exciting poll that we held with some very interesting questions. I've shared it with people. Can people see that?
Yes.
Yeah, and I don't think there's much to be said about it. I just say it's rather sad to see the end-all division didn't really resonate with people, but if you look at the graph, it probably reflects the severity of each extinction, which is rather interesting. Then obviously one of the key questions, yes, the future of this planet is in our hands. We are probably going to cause, I think undoubtedly we are causing the next extinction. And then there were some other questions about the geoscience community and communication, and yes, maybe we're doing well. I suspect probably not. And then survival of the fittest, the most adaptable, and then finally, what are you actually going to do when you're facing an extinction event? Well, I think other wins it with 45% rather than hide in an underground cave or run to a high latitude area or learn to dive. So look, thanks everybody. Thanks for all of you. I think we've still got 55 people hanging on. We had 130 people at the peak, but thank you all for staying the course. And I think the discussions have been, you know, quite interesting and stimulating. I thank Mike for leading that and keeping everybody on the page with these things. All I can say is thank you very much to all the speakers, and I wish you all a very good weekend. Thank you.
Thank you. Thanks all.
Thank you.
Thank you. See you.
Thanks.
Have a nice day.
Bye.
Yeah.
And a nice weekend.
Yeah.
You
Thank you.
too.